talk awards by Professor Stanley Wu and Professor Ralph Olemüller, and I request Dr. Rajshekhar to kindly chair this session.
Thank you, Niti, and sorry people are still on the way, but we have to start at least. Dr. Stanley Rue's talk is now. Unfortunately, because of his family situation, health situation, he could not come, but he was kind enough to send us the greetings. But who is Stanley Rue? Many of you people may not be aware of it. Dr. Stanley Rue is a very distinguished professor in the University of Texas. He is a Yale graduate for his PhD, and he has worked extensively on signal transduction with phytochrome, and also gravitational biology, using the extracellular, exploring the extracellular ATP signaling, and also photomorphogenesis in general. When he was working with gravitational biology, this work was very impressive, so his work was even accepted to be presented in NASA research projects. I think apparently they sent their materials to the space as well. So he is very distinguished faculty, as well as AAAS fellow, ISPP fellow, also as well as ISPP Excellence in Education Award in 2015 he received. So essentially his primary work is on the ATPases and also A pyrase, that is D and TPases in regulating plant growth and development. Why Stanley Rue we chose? Dr. Sudhir Sapori, he has worked with him when he was on sabbatical from JNU. He worked with him. At the time I suffered a little bit. One year he was not there, and then I was down to work on also Cipra was there, just fine. But Dr. Sapori was continuously kept in touch, even remotely. So he was sending us what experiments to be done and all these things. So I really enjoyed. So at that time he told me two important things, Dr. Sapori. Person who climbs on other person's ladders, they fall one day. And another thing, you have to choose in the life, you want to be a king by following Akbar, maybe Ralph doesn't understand, one king of India, and or by Aurangzeb. In both the ways you can become a king and feel happy that you are a king, but how you became king is very important. That is what Dr. Sapori taught me. So that is one of the things. Later on I will tell some other things. So now we are going to play Dr. Stanley Rue, who kindly agreed to record his things. He was very much sad that he could not attend. Originally he was interested to attend and present these things. Can you start? Yeah, video. No, video of the lecture.
Hello, my name is Stan Rue, and I want to begin by thanking the organizers for inviting me to this symposium honoring my friend and colleague, Professor Sudhir Sapori. He is very deserving of this honor, not only because of his significant scientific accomplishments, but because he is such a kind and compassionate man and a great mentor and a great leader. I first met Sudhir when he came to my laboratory in the 1980s to study a process called signal transduction in plants. It's a process by which plants convert input environmental signals into adaptive responses. At that time, he and I were both interested in two principal signaling agents. One was phytochrome, a photoreceptor that turns on light responses in plants, and the other was calcium, which is a kind of an amplifier of many different signaling events. As I was preparing this talk, I realized that the research Sudhir and I did together, as well as our conversations, led me to plan some experiments which resulted in my discovery of the enzyme, which would be the main topic of my talk today. That is the enzyme A pyrase, which is an NTP-dease. So let me now talk about this story about A pyrase. It's a story of unexpected interconnections. The connection of A pyrase with phytochrome, with auxin, a growth regulator, and with drought stress, which definitely affects growth. I would now like to make the connection between the research that Sudhir did in my laboratory and the research we did together that led to the A pyrase discovery. If you look at this slide, you will notice that in the two publications that we did together, one was on calcium and phytochrome, and one was on calmodulin, which is a calcium binding protein. So when I thought about this research, I realized that the interest in phytochrome at that time was mainly phytochrome regulation of gene expression. That is, it functioned in the nucleus. And so he and I talked about this a little bit, and I began to wonder, were there any nuclear proteins that were regulated by both phytochrome and by calmodulin? And it turned out that in 1980 there was a PhD dissertation that was published in Mark Jaffe's laboratory, which indicated that you could isolate nuclei from P and show that there was an ATPase in nuclei that was red light activated and far red light inactivated, which is an indication that it was phytochrome regulated. Phytochrome responses are turned on by red light and then turned off by far red light. So this dissertation said there was an enzyme in nuclei, in Ps, that could be turned on by red light and turned off by far red light. So we decided to look to see more about this ATPase, and the next two slides show that Yuru Chen in my laboratory discovered that, in fact, there was an NTPDase, or a pyrase, nucleoside triphosphatase activity in isolated nuclei that was photoreversibly regulated by light. We published that in Plant Physiology. And then the next year, she went further and isolated nuclei and purified this enzyme from nuclei. And that was published in Journal of Biological Chemistry. It showed the purification and partial characterization of a calmodulin-stimulated NTPDase. So here we had made the discovery that we were kind of looking for, that yes, there was an enzyme in nuclei that was regulated both by phytochrome and by calcium and calmodulin. Okay, so let's talk a little bit more about these apyrases, NTPDases. We made the discovery of them in P nuclei, but then the Arabidopsis is a model system that is much more amenable to biochemical analysis. So we switched to look at apyrases in Arabidopsis. And this is what we found out. First of all apyrases, which are very highly conserved throughout evolution, from algae and humans all the way to higher plants. So all of these apyrases remove the terminal phosphate from NTPs and NDPs, but not NMPs. So they're different from acid phosphatases and alkaline phosphatases in that they cannot remove the terminal phosphate from AMPs. There are 7 of these in Arabidopsis, but only 2 of them seem to be strongly associated with growth regulation, and we call them apyrase 1 and apyrase 2. And it turns out these 2 are almost duplicates of each other. They're 87% identical. If you knock out only one of them, you don't see a phenotype much different from the wild type. But if you suppress both of them, you have a very significant impact on growth. So suppression of both apyrase 1 and 2 dramatically inhibits the growth of pollen, of hypocotyls, and of roots. And if you overexpress them, that enhances the growth of all of these tissues. We did genetic and chemical analyses of these apyrases and found that they hydrolyze extracellular ATP, and we call that EATP. And thus they were able to limit the concentration of extracellular ATP. So that meant that they could function, this apyrase could function in the ECM, the extracellular matrix, or the wall. However, it turns out that apyrase 1 and 2 also localize in nuclei and in Golgi. And we can assume that it could regulate the concentration of ATP in any of these locales. Okay, so if we say that some of the apyrase functions in the wall and that its main substrate is ATP, that raises the question of, is there any ATP in the wall at all of plants? And if so, how does it get there? And this slide indicates the different ways in which ATP could be released into the wall. Obviously, if you wound a plant cell and break the plasma membrane, then the millimolar ATP that's in the cytoplasm will leak out into the wall. There are MDR transporters that can move ATP from inside to outside. These are common both in plants and animals. It turns out if you touch a plant cell and touch stimulate it, it will release ATP to the outside. Osmotic stress, which certainly moves the membrane, also releases ATP to the outside. And then there's the process of secretory vesicles. So it turns out that both in plants and animals, these secretory vesicles are actually kind of loaded with ATP. In animals, they've been measured as high as millimolar ATP within the vesicles. And when these vesicles fuse with the plasma membrane and dump their contents into the wall, which is a process that happens during growth. In order for the wall to expand, you need the wall components. Then this fusion of secretory vesicles to the wall will release ATP into the wall. Okay, so once ATP gets out there, does it do anything? Yes, there's the case in plants as in animals that almost all sensory responses depend on a receptor of the stimulus, and there are receptors for extracellular ATP. In animals, there are two types. One is called P2X and P2Y. The other is P2Y, and I think last year there were over 500 papers published on P2X and P2Y phenomena in animals. So it's a major topic in animals. In plants, it was much later before the receptor for ATP was discovered, and that was discovered in 2014 in Gary Stacey's lab, and it was initially called DORN1, now referred to mainly as P2K1. This was the first receptor discovered in plants. And it is definitely involved. It was shown, even in the first paper in Science, that it is related, it mediates calcium-related responses in to extracellular ATP. But it is not the only receptor in plants. So the note below, I say that those ectoapyrases, that is, apyrases that would function in the wall or outside the cell, would regulate the extracellular ATP because they could hydrolyze. ATP is a substrate. It would break down that ATP basically to AMP, and thus it would modulate plant responses to the extracellular ATP signal. So you could not activate a DORN1 unless there was ATP out there, and if you want to regulate the concentration of extracellular ATP, then apyrases could play a major role in that. So we were interested in the role of apyrase in regulating growth, and so we raised the question, are ectoapyrases, that is those that function in the ECM, are needed to sustain growth? And I point out in this slide that, as I indicated earlier, that one of the ways ATP gets outside the cell is through secretory vesicles. They deliver proteins and carbohydrates to the expanding cell wall, and they release ATP. So this happens in every cell that's growing. In order for it to grow, it's got to have these secretory vesicles dump ATP into the wall, and so this increases the extracellular ATP. We found out that, maybe unexpectedly, high ATP inhibits auxin transport and growth, and we'll tell you a little bit more about that in the next slide. But if you wanted to therefore limit the concentration of extracellular ATP, you would need an enzyme to hydrolyze that extracellular ATP, and it turns out among all the phosphatases that can hydrolyze ATP, the strongest one is a pyrase. It has the lowest Km for ATP hydrolysis. So it's maybe not too surprising that if you suppress a pyrase expression, this will raise the extracellular ATP, and that increase in ATP could inhibit growth. Correspondingly, it turns out that cells that are growing very rapidly, maybe not surprisingly, have a very high level of a pyrase, which would play the role of limiting the increase in extracellular ATP as these rapidly growing cells grow. So here is the evidence that the increased extracellular ATP will inhibit polar auxin transport. This was published by Tong et al. In 2003. You can see that as you raise the concentration from 0 to 1 millimolar, you see significant inhibition of basipital auxin transport, 2 millimolar even more, and 3 millimolar even more. Now, how high would the extracellular ATP get in cells that are growing? We have some evidence that it does get fairly high using some ATP sensors. Whether it would stay at 1 millimolar very long, we don't have the kinetics of that. But for sure, as you raise the extracellular ATP, there would be, if you will, the side effect of inhibiting auxin transport, which of course would inhibit growth. So if you wanted to now test more rigorously the role of a pyrase in regulating growth, a very good system to use would be phytochrome regulated growth changes that happen as the seedling emerges from darkness into light. So it turns out that in etiolated seedlings, that is dark grown seedlings, hypocotyls grow very rapidly, but roots and cotyledons grow slowly. In other words, the plant is allocating its resources to growing the hypocotyl. Basically, it will enable the plant, the seedling, to get to the light faster if the hypocotyl is growing faster. But when it emerges into the light, the phytochrome activation by red light inhibits the hypocotyl growth and stimulates root and cotyledon growth, which means that at this point, the plant is, if you will, deciding that there's no need to hurry growing into the light. Once you're there, now you can reallocate your resources to expanding the cotyledon for photosynthesis and helping the root grow faster. So then this raises the question. Now you have a phytochrome system that has opposite effects on two different tissues. Hypocotyls would grow fast and would slow down when you activate phytochrome, and roots and cotyledons would grow slow in the dark but then expand in the light. So then the question would be is there a correlation between a pyrase expression and growth changes during the de-etylation process? So we did this study in a number of different ways. One was to use promoter GUS to assay where were the transcripts of a pyrase most strongly expressed. If you look in dark-grown tissue, you see that very strong expression in the hypocotyl. Very little expression, you see very little blue color in the cotyledons. But once you give light, once they grow into the light, you see almost no signal, no promoter GUS signal in the hypocotyls, but very strong signal in the cotyledons. So what this means is that the expression of a pyrase, at least the transcript expression of a pyrase, is strong when the hypocotyl is growing rapidly and not very strong when it is not growing rapidly. So to further establish this phenomenon, we looked at phytochrome mutants, where if the mutant does not have phytochrome, then light doesn't affect its growth because phytochrome is the photoreceptor that mediates the light effect. And so what we see in phytochrome mutants is that the high expression of a pyrase in the hypocotyl remains strong even in the light. and the growth remains strong in the light because the plant is acting as if it's still growing in the dark if it doesn't have phytochrome. Even more interesting to us was the fact that the kinetics of loss of a pyrase protein, and we were able to use Western blots to look at the protein loss, follow the same kinetics as the kinetics of decreased growth of the hypocotyl. So on the left you have some results from a laboratory in Wisconsin that showed that it is about a 7 or 8 minute lag between the time you turn on the light and the time the growth rate of the hypocotyl begins to slow down. And if you look on the right, what you see is in the Western blot, if you time how long does the strong expression of the protein in the hypocotyl remain, it's basically severely decreased by 7 minutes and almost not there at all after 7 minutes. So the light-induced loss of a pyrase protein in hypocotyls parallels the light-induced growth inhibition. So that kind of implies that a pyrase could be really important in this response. So now let's make the auxin pyrase connection, beyond just saying that high ATP inhibits auxin transport. What about a pyrase? If a pyrase is controlling extracellular ATP, then maybe there is a connection. So you remember in 1937, you remember, I'm not even old enough to remember that, that without auxin there's no growth. So then the question is, if without a pyrase 1 or 2 there is no growth, then does a pyrase 1 and 2 expression affect auxin production or transport? That's the question. So we collaborated with Gloria Moudet to do auxin transport measurements with radioactive auxin, and these are the results that we found. And what we're looking at is the clear bars are the percent of polar auxin transport through the etiolated hypocotyls, and the dark bars show the effect of NPA, which is an auxin transport inhibitor. So in every case, the dark bar, which is NPA treatment, blocks auxin transport, as shown many times in the literature. But if you look at the clear bars, what you see is that there is no effect of adding estradiol to the wild-type seedlings. Why do we say that? It turns out that we used a construct to inhibit a pyrase 1 and 2. The construct was like this. It was negative, it was null for a pyrase 2, but then the pyrase 1 could be partially suppressed by turning on an RNAi construct in a mutant. So we used a mutant which we called R24A, and in that mutant, a pyrase 2 was null, and a pyrase 1, without the RNAi being turned on, was still there, 100%. But once you gave estradiol to turn on the RNAi construct, then the mutant, R24A mutant with estradiol, had no apirase 2 expression and only 40% of the normal apirase 1 expression. And in that case, what you see is in R24A plus E, you see a very significant inhibition of auxin transport. In contrast, if you overexpress a pyrase, this OE stands for constitutive expression of a pyrase, what you see is that the growth, the polar auxin transport and the growth are enhanced. So this is very strong evidence that the expression of a pyrase, the level of expression of a pyrase, either suppression or overexpression, correlates very well with the rate of auxin transport, which would help us explain why a pyrase regulates growth. It would do so through auxin. So not only does auxin apyrase expression affect auxin transport, but it also induces major changes in gene expression. And when we did RNA-seq to assay the gene expression changes that happen when you suppress apyrase, what we found is that the most significant gene expression changes induced by apyrase suppression are in genes that are regulating wall composition and extensibility. So for a cell to grow, the wall has to be extensible. And you can regulate growth by regulating those enzymes that control extensibility of the wall. And among those enzymes, one of the most important is the peroxidases. And what we found is that when we suppressed apyrase, 5 different wall peroxidases were strongly upregulated when apyrase 1 and 2 were suppressed by the R2,4A construct. That is, you had null for apyrase 2 and suppression of apyrase 1. And these included peroxidase 15, 49, 52, 53, and 54. And their upregulation ranged from 21.6 to 2.43. All very statistically significant. And what we also found is that when these apyrases were upregulated, that these peroxidases were upregulated, we had very strong changes in the wall flexibility or the wall extensibility. It was decreased significantly. So there was a correlation between the upregulation of these peroxidases and the stiffening of the wall, which would certainly account for why the cells weren't growing very well in the R2,4A mutant. Okay, so up until now, all of our data on apyrase and phytochrome showed that if you suppress when you suppress growth, the apyrase level drops. So this would suggest that you need apyrase expression in order to sustain growth, because if you get rid of the apyrase, then you see decrease in growth. However, as we indicated earlier, in the case of phytochrome, different tissues respond differently to the red light stimulation. The hypocotyl is inhibited by red light, but the hook cotyledon, the hook expands and the cotyledon expands, and the roots grow longer in red light. So during deetiolation, hypocotyl growth is inhibited, but root and hook cotyledon growth is enhanced. So this raises the question. We know apyrase is involved in the inhibition responses to phytochrome, that is the hypocotyl response, but is apyrase expression also needed for the phytochrome-induced enhanced growth of root and the hook cotyledon tissue? This question was investigated by a grad student in my lab, Gayane Wirarante, and we published those results in a 2022 issue of Plant Physiology. So let's review those results. If you suppress apyrase expression using R2,4A suppression, what you see is that within the period of time that you see a strong increase, normally you see a strong increase in cotyledon expansion within 24 hours. So at 24 hours, what you see is that the wild type, WS, at 24 hours is growing, is expanding, has expanded already to some extent. But at that time point, at 24 hours, whether you have added estradiol or not, you see inhibition of that expansion. What this means is that even if all you have is suppression of apyrase 2, no effect on apyrase 1, you still see, that is R2,4A minus E, you still see the inhibition of expansion of the cotyledon. But if you now add estradiol, you see the inhibition is even greater. So if you go out to 72 hours, the difference between C and B, that B is when you only inhibit only have inhibition of apyrase 2, but C, you have inhibition of both apyrase 2 and apyrase 1. You see a very strong inhibition of expansion of the cotyledon. What about the roots? So this result shows that suppressing apyrase expression also inhibits R-induced increase in root growth. So if you follow the A's, that is the wild type expansion of root length over time, starting at 0 hours going up to 72 hours. If you follow the A's, you see the bar gets longer and longer. B is if you only have R24A without estradiol, you see some inhibition, so apyrase 2 is involved in this response. But if you add estradiol, what you see is that there is no expansion of the root all the way up to 72 hours. In other words, if you have apyrase 2 knocked out and apyrase 1 suppressed by 60%, the root basically doesn't grow. So that's a very strong indication of apyrase involvement in mediating the light response on root growth and also, obviously, as we said before, on cotyledon growth. So what about the kinetics of this change of the amount of apyrase present in the root? Okay. I don't know if any of you are working with Arabidopsis, but can you imagine isolating a gram of Arabidopsis roots, which it took a lot of time. Anyway, this graduate student was able to do it. But if you follow the WS on the far left, you see the level of apyrase is reasonably strong in the root. If you knock out apyrase 1, you see a great decrease in the amount of apyrase in the root, indicating that between apyrase 1 and apyrase 2, there's more apyrase 1 in the root than apyrase 2. So if you knock out apyrase 1, the level, the Western blot signal for apyrase goes way down. It goes down a little bit if you just knock out apyrase 2. But now if you switch and look and see wild type after red light, you see a very strong increase in the signal of apyrase in the root. And if you want to look at what happens if you knock out apyrase 1 or apyrase 2, you really need both of them in order to have the increase in root growth and the increase in apyrase expression. So what this says, and by the way, this was measured at 24 hours, and by that time the root has a very strong acceleration of growth, and in parallel a very strong increase in the level of apyrase expression. Okay, so what about this process of the hook opening up? And on the right, in the upper right-hand corner, you see an image of an etiolated seedling of Arabidopsis in the dark. The hook is bent down, and it has that strong hook shape. So that's the normal situation in the dark. If, however, you overexpress apyrase, what you see is that even in the dark, this overexpression of apyrase begins to make the hook open. It's acting as if it's in the light, even though it's still in the dark. So this is an indication that apyrase is playing a major role, not only in root regulation, but also in the opening of the hook and the expansion of the cotyledon. So about 5 years ago, some scientists studied this process of hook opening and cotyledon expansion, and they found there were two SAUR, S-A-U-R genes that were critically involved in this phenomenon. SAUR is small auxin upregulated gene. And if you look in the dark, what you see on the upper image, you see that when you give red light to make the hook open, you have a strong upregulation of SAUR50 and a strong downregulation of SAUR17. It turns out that SAUR17 has to be downregulated in order for SAUR50 to be upregulated. So this phenomenon is well described in the literature. So we wanted to know what happens when we see a pyrase promote hook opening in the dark. So we looked in the dark, and what we found is that SAR50 is very strongly upregulated even in the dark, and SAR17 is downregulated even in the dark. So this B panel here explains why in the dark the hook cotyledon opened even without light. So again, a strong indication of a pyrase being a mediator of this phytochrome response. In addition to the SAR gene regulation, we also noticed, again, a peroxidase wall gene regulation. And here we go a little bit further than we showed before. Before, we only showed what happens when you suppress the pyrase. You see upregulation of peroxidase 15, 49, and 59. We already showed you the data on the far right column. But what happens when you overexpress a pyrase and you promote growth? Turns out when you promote growth, you downregulate these same peroxidases that are upregulated when you inhibit growth. So this really links a pyrase regulation of wall peroxidase gene expression as a critical step in the effects of a pyrase expression on growth. Okay, so beyond our Arabidopsis studies, which strongly indicate that pyrases are critically involved in growth control, using phytochrome as a model system to study that, we wanted to know whether this had any agricultural value. And so we began expressing a pyrase in crop plants. And what we found is that the constitutive expression of a PA pyrase in soybeans significantly expands the root system architecture and drought survival in soybeans. So we link the drought survival to an expanded root system architecture. And a lot of people have shown that connection. So the connection of a pyrase with drought control is it regulates the expansion of the root system architecture, and that allows the plant to be more resilient against drought. And it survives better if you overexpress a pyrase. So if you also overexpress a pyrase in soybean, in field-grown soybeans, we have a very significant 15 to 25% increase in yield of the soybeans in the field. And this scientific reports study showed results of multiple field trials over multiple years, all with the same result. If you overexpress a PA pyrase, you get greater yields, significantly greater yields. So we think that understanding a pyrase's control of growth would have some benefit for agriculture. So the last thing, in terms of data, is raise the question of where is a pyrase working when it's doing all these things to control growth. And we think it can function in any one of three locales. If it's functioning in the nucleus on the far left, you could imagine that it could change gene expression. In fact, Yuru Chen, when she isolated a pyrase from peas, she found that it co-isolated with chromatin proteins. So we think one of the chromatin proteins in peas is in fact a pyrase. If it's on the plasma membrane, it could be anchored in the plasma membrane with its active site facing to the outside, and in that way it can regulate extracellular ATP concentrations, and that has been shown by many people to have an impact on growth and seed yield. And finally, if it's operating in the Golgi, which is another place it's been found, it would have an impact on protein glycosylation, because the ATP-ADP equilibrium in the Golgi plays a critical role in whether or not in the process of protein glycosylation. And that would, of course, play a big role in which proteins go to the plasma membrane and function there. So we think it can function in any one of three locales and still have an impact on growth. So here's kind of a summary and implication of our findings. The suppression of a pyrase 102 and of auxin transport may be an early step in PFR-induced growth inhibition of hypothalamus. Similarly, upregulation of APARase 1 and 2 expression and of auxin transport may be an early step in the phytochrome PFR-induced growth promotion of roots and hook and cotyledons. And finally, the ectopic expression of a PA pyrase in soybean expands its root system architecture, enhances its drought resistance, and its seed yield. So it has significant ag value. Of course, there are many unanswered questions in our research, and we're still active in doing research. We still have grant money from Texas Crop Science, which is a company interested in commercializing a pyrase in crops. But what we are studying now is how are the early signaling steps, that is calcium increase, transduced into changes in the transport of the growth hormone auxin. That is, how does a pyrase regulate auxin transport? Is the nuclear or the ECM function of a pyrase more critical for growth? And what we're doing there is we are knocking out the nuclear localization signal and looking to see how that mutant functions, and we're knocking out the signal peptide, which would keep it from going to the ECM, and seeing how that functions. So that work is in progress. And we also want to know, we know that a pyrase in the nucleus does bind to DNA, because Yu Chen was able to show that it binds to a DNA affinity column. But it also seems to be a chromatin associated, so maybe it could function in the nucleus either through its DNA interaction or through its interaction with other chromatin proteins. And then we want to know, with respect to calmodulin activation, what cellular proteins other than calmodulin interact with a pyrase in the nucleus, Golgi and ECM, to enhance its function. We're doing yeast 2 hybrid and co-IP to look at this. So here's the team that did all the work. I just pushed papers around, and cheer and that kind of stuff, but these are the guys that actually did all the work. Greg Clark is the manager of the lab and is a co-PI of all our grants. Mary Salmi did some of the early work on the pyrase disappearance after red light and hypocotyl. Rupa did the field trial studies on soybean. Monostripathy did some really beautiful work showing that if you inhibit a pyrase, this can have an impact on some agents that are involved in attacking, like fungi. Minhee Lim did the RNA-seq studies on changes in gene expression. Jichul Nam is in my lab now. He's doing the co-IP. Tanya Sabaral did the 2022 scientific reports work. Ashley Cannon has studied a pyrase in ferns. Jing Yang showed a connection between a pyrase and annexins. Gayani Weeraratne did the red light effects on root and hypocotyls, and the cotyledon and hooks. And then we have many undergraduates doing research in our lab. We also had several collaborators, which are listed on the right. Okay, that's what I have to say. Thank you very much for your attention. And I would welcome, if you want to, I can't answer questions, obviously remotely, but I am very open to you just sending me emails, and I'll be happy to answer any questions you may have. Thank you very much. And once again, congratulations to Sudhi for an amazing career and continued contributions to the advancement of science and plants. Thank you.
So from any of you guys have any questions, like he said, you can send directly to him, email to him. He may be able to respond. But when I'm looking at it, I wish maybe many of us should be wishing, like in his age, we should be that much active. But perhaps it's amazing how much active he was. Yeah, lots of he memorized completely. He's not looking at the paper also. So the whole thing is going on. Nice. So now next speaker will be Rolf Weinmuller, and I will invite him to come to the podium. Are you ready, Rolf? Yes, good. So Rolf Weinmuller comes from Germany. He did actually northern part of Germany, Munster, and he came to Freiburg to join Hans Most lab as a PhD student. It was coincidental. When I went as Humboldt fellow, he joined at that time. I think he was already a fellow, PhD fellow. It was a great interaction we had, And also what was interesting is that at that time there was a very hot topic. Rolf is significantly responsible for this plastic signal development signaling thing. Then I had an opportunity to participate, and hence more also kindly included me into that program. So I was an author of that paper, which is very well cited even now. So I'm very proud about that. Then it was so incidental. Winslow Briggs was also there at that time in a sabbatical in the same lab. So it was amazing for me, especially. I will tell about it later on. But Rolf had a good interaction with him. So he had, after finishing PhD in Hans Moos lab, he had an opportunity to work in abroad in Winslow Briggs lab on a chromatic adaptation of algae. Most importantly, Rolf has achieved his PhD with the highest, what is it called, summa cum laude or something. So that is highest award. Means one has to do outstanding work, we will get PhD. PhD is a grade in Germany. At the same time, it's an interesting time. And only PhD is not enough to become PI. You have to do some other extra studies, and it's called habilitation. You have to do that. That is what Rolf did when he came abroad to Winslow Briggs, a portion of it. Later on, he returned back to Munich, and he joined as independent scientist, but he has to work in a group. But everything has to, money and all has to come from his own writing. But it was an incidental. My boss, Dr. Sudhir Sapuri, I happened to be a Humboldt fellow in his boss group, I think, Herman's lab. So that is how Dr. Sapuri and Rolf interacted together. So it is a nice interaction. If I look at it, that I know Rolf and Rolf knows my boss, like that, whatever it is. It's amazing. So here, what he did is that he worked on. Later on, he moved on after completing habilitation in Munich. He moved to Jena to work on plant microbe interactions. Also with the Max Planck Institute in Jena, he collaborated and worked on the Max Planck Institute to work on chemical ecology of the maybe algae interactions or something. But anyway, he is the most successful scientist. Very important, he is very jovial. He works very hard. By evening, he relaxes nicely, going to the beer and all. He used to drink amazingly. Once me and my wife visited him, and my wife was surprised. I never saw such a German guy drinking so much. Full bottle will finish it up. But importantly, I am very happy to learn he stopped drinking. So he doesn't drink anymore, but he smokes a little bit. That's it. It is very nice to have him to talk today. Please come on.
Okay. Yeah, thank you very much for the nice introduction, and it's a great honor for me to be here today on the occasion of the 75th birthday of Sudhir Sapuri. I wish you all the best for the future. I wish you all a nice day today. And as far as if I remember correctly, we first met in 1992 or 1993, early 1993 in Munich. This is the Botanical Garden in Munich, in the laboratory of Professor Hermann, and actually we came very close together, and there was another. What do you want? Okay. We came very close. We cooperated a lot in the lab, and there was also another visitor at that time in Munich, Professor Akhilesh Tyagi, and we all cooperated a lot, not only within my group but also with other members in the laboratory, and looking at this. This is the output of the stay of Professor Sapuri in Munich, and he published probably more, but these are only the papers that he published together with me. And it looks very funny when I look at this list of publications, but it was very important at that time for me because I was in the process of getting the habilitation, which is necessary in Germany for becoming a professor. And you can clearly see here from this huge number of papers with contributions from Sudhir, that this was a substantial contribution to my career in Germany. So we became great friends and kept our friendship until now, and thank you very much for this, Sudhir. But at that time I'd never been in India. And he told me all the time, I have to come to India. And the first time that this was possible is when he organized a conference on signal transduction in plants, and that was exactly happening here in this room, if I remember correctly. Yeah? And this was the first time that I came to India in September 1999. And we can also prove that a little bit since, I forgot to mention, these yellow people which were co-authors on the papers, I still have contact to them, and they wrote emails to me in the last maybe 2 months. And I should tell also all the best to you from Thomas Oberstedt. He's professor in USA now. Cordelia Boll is professor in Munich. Viktor Kuznetsov is professor in Moscow. And Irina Sherameti is working in a company in Jena. So, this was the outcome of the conference that Sudhir organized here. Everybody knows that, on signal transduction in plants in 1999. But after this small introduction, I would like to go on a little bit with the activities in my laboratory. And it proceeds with the introduction from Stanley Wu. He was talking about calcium, he was talking about cell wall architecture and how that can be modified during growth and development. And basically, I'm looking at it from another aspect. And I will talk about similar aspects that were already introduced by Stanley Wu. What we are trying to do is to understand how plant-microbe interaction works. We take our Arabidopsis as a model system, and this was all done by Indian students and Indian visitors. They all introduced this whole story and initiated this whole story in our lab. Today I'm talking about a story that has been done by a non-Indian PhD student, a person from Taiwan, but all the work leading to the work of that what I'm presenting today was done by Indian students and continued also by Indian students. So the idea was to understand how can roots monitor what's going on in the environment. Are there beneficial microorganisms, pathogenic microorganisms? And a very crucial aspect is the cell wall. The cell wall has to be intact, and when the cell is growing, there must be modifications in the cell wall. That means internal signals must degrade cell wall polymers and then rearrange the cell wall polymers to new polymers. And when pathogenic or friendly microorganism attacks a root in the soil, then of course there will be cell wall degradation. And the cell wall degradation, as well as the remodeling that is required during growth and development, that clearly must be monitored by the cell. People have recognized it many, many years ago, but still we don't know exactly how that can be recognized. So in order to understand what's going on, as you know that this is the architecture of a cell wall. Here is a plasma membrane, and you have at least 3 different types of polymers, polysaccharides in the cell wall. This is cellulose. I'm only talking about cellulose today. We have pectin and hemicellulose. And cellulose is different from the other two because this is more than 50% in the primary cell wall, and it's a very strong polymer, and you can see that nicely because it forms a crystallized structure, different from pectin and hemicellulose. And breaking down of the crystalline structure is very difficult, at least enzymatic breakdown is very difficult. So cellulose is a sugar polymer, beta-D-glucose moieties polymerized, and it's very simple compared to all the other sugars which are needed to build up pectin and hemicellulose. And if the cell wall is broken down and cellulose is broken down, then you get oligomers or monomers of these cellulose. And what is normally happening is cellulose degradation occurs by endogluconases or exogluconases, and normally you get dimers or C4 or C6 compounds, which can then be further cleaved by glucosidases to glucose. So this glucose is the end product, of course, and that can be used as an energy source for the microorganisms. Of course, that is very clear. But everything else, the dimer and polymers or oligomers with N3 and 2N7, they have signaling functions in plants. This difference is very important. Only the completely digested cell wall, the glucose, normally functions as an energy source, whereas the other compounds function as signaling compounds. And when you look at a biological system like a rhizosphere, for instance, you find mainly C2 breakdown products, C4, C6 breakdown products, so cellobiose, 4 and 6 sugar molecules. You find little C3, C5 or C7, but C3 is very important because there is a sensory system for C3. And also different microorganisms use C3, so that means 3 glucose moieties, cellotriose, as a signaling compound to interact with plant roots. Yeah? So C2 to C7 has signaling functions, whereas the complete breakdown products, glucose, has no signaling function, but functions as an energy source for plants. So how can the cell wall be broken down? Of course, there are plant enzymes, and almost all of the microorganisms, bacteria, nematodes, fungi, all of them contain a huge variety of different enzymes which can all degrade cellulose. I give you only one example. These are the lipid polysaccharide monooxygenases, which become very popular for different reasons, present in many, many different microorganisms, in particular fungi, and they are also capable of degrading oligomers with crystallized structure. So this can be a very efficient way of microorganisms to degrade cellulose, for instance. Not only cellulose, but also cellulose. So at the beginning of the whole story, we found out that a beneficial fungus, Piriformis spora indica, which was brought by Professor Arjivama to our lab, induces cytoplasmic calcium elevation, and this was found out by Jyoti Vadaseri in our lab. So when we discovered this, all students in my lab wanted to work on it. It was very difficult to continue with my old research. Everybody was interested in this plant microbe interaction, and I also got more and more funding for this, and therefore we try to understand how this can be possible. So there was an accumulation of cellotriose in the apoplast of the microorganism, and in this case Piriformis spora indica, and somehow this is perceived by the root cell, and the first response that we can measure is an increase in cytoplasmic calcium elevation. So we then checked whether the different other cell oligomers can also induce cytoplasmic calcium elevation, and what I can show you here is the cellotriose form is the most active one, or the other forms are less effective, and as I told you already, the monomeric form, glucose, is not effective. So we wanted to understand what this calcium is doing. Of course, this is dose dependent. The next important finding was that the response is mainly happening in roots. So what we find is when we apply cellotriose to the roots, then the signal is much stronger than application to the shoots. So if we think about perception systems, then it appears that there must be a specific perception system, or let's say a receptor, is present in the root and to a lesser extent in the shoot. Okay. So we then were interested in understanding, is this calcium really important, or is it a side effect, or for what do we need this calcium elevation? And the result of all the studies is now summarized in this sort of cartoon. Downstream of calcium elevation, we have a number of other responses which require this calcium elevation. First of all, there is increase in apoplastic ROS, reactive oxygen production. And this calcium elevation is very fast. It occurs after 30 to 90 seconds. It's a very simple indication whether there is an interaction, for instance, of microorganisms with the roots, because you can measure that after 30 to 90 seconds. And a little bit later, you find an increase in the production of reactive oxygen species. The next effect that we can easily see is, and that occurs 1 minute later after the calcium increase, is membrane depolarization. And if you take the calcium increase, the ROS production, and the membrane depolarization together, then we can propagate the signal. And this is very important. If we start an interaction or a disruption of the cell wall at one place, for instance in the root, or the other tissue must be informed about it. For instance, there must be an information transfer from the roots to the leaves. And this requires a systemic signal propagation. And the systemic signal propagation requires a combined action of calcium, ROS, and membrane depolarization. What is also very well known and often investigated in different laboratories, that there is a signal transduction pathway leading to ultimately changes in gene expression. And this changes in gene expression occurs mainly via MEP kinase signaling 3 and 6, which become phosphorylated. And then here are examples for a number of genes which are upregulated. Often people are looking at defense genes, but this is not the only category of genes which are regulated. So what I wanted to show you with this cartoon is that all starts with calcium, and this calcium appears to be very important for all the downstream events. And if you kill this calcium response, maybe all these downstream responses are gone. And cellulose repair, I'll come to that in a moment. So our idea was to generate mutants in Arabidopsis which do not show calcium elevation. And if this is the case, if we have no more calcium elevation, we can see all the downstream responses. So what we did is we used cellotrios and looked for mutants which do not show calcium elevation anymore. And we isolated a number of different mutants. I talked today about one mutant which we call CORK, cello-oligomer receptor kinase mutant, which we then identified by the classical techniques, ultimately Illumina sequencing and map-based cloning techniques, confirmation by complementation assays and so on, all the molecular techniques which are available for Arabidopsis. And that's a new malactin-containing receptor kinase, which I would like to describe you a little bit more into detail. The studies were done by the Taiwanese PhD student Yuan Zhang right now. Here's the architecture of this protein, and I will guide you through the architecture in the next few slides, showing you this again. It's a membrane protein. The membrane protein is, of course, has a signal peptide, is transported to the membrane, has a leucine-rich repeat sequence that is required for, or that is involved in protein-protein interactions in the wall. Then there is a malactin domain. I'll tell you more about this malactin domain. This all is in the apoplast, and here's the transmembrane segment. And within the cytosol, you find this kinase domain that should be possible, that should be activated here to induce signaling. In order to test functionality of this protein, this receptor kinase, which has not yet been investigated before in Arabidopsis, to our surprise, almost every gene has been turned around 200 times in Arabidopsis, but nobody has looked at this gene yet. So we could first show that the signal peptide is working and that the fusion protein ultimately ends up in the plasma membrane, here labeled with GFP. So we can at least conclude from this experiment that the secretory pathway for this protein is working, and the protein finally ends up in the plasma membrane. We wanted to know whether the protein is active and able to induce transcriptional changes. For this we used a protopast luciferase assay system after application of cellotrios. So what we did is we applied cellotrios to the system. Here's a positive control with PEP1, so you can see that the transcriptional activity is increased in the control. Here's the negative control, and if we give cellotrios to our construct, we can see that there is an activity. So the receptor kinase is active in this assay system, so that we can conclude that signaling occurs also in that mutant. Next, we wanted to see whether the kinase activity is active. I don't want to go into details, these are all controls, but you can see a phosphorylation of mycelium bovine protein after stimulation of CORK1 by cellotrios. And here are several controls showing that the kinase activity is at least functional. So if we have a kinase activity, that means there must be a phosphorylation event. And then I went back actually to the literature, and this cartoon probably will be a nice introduction for the next few minutes of my talk, in which I try to tell you what this protein is primarily doing. So it doesn't matter whether you look at animal systems or plant systems. There's a signal from the outside that is somehow perceived by a receptor kinase. In our case, it's the cell oligomer or cell triose. You have a number of interaction partners, and at least in our case, it's very clear that there must be calcium channels, or at least one calcium channel involved, since we can measure cytoplasmic calcium elevation. And this occurs within about 90 seconds. So this calcium interacts in this calcium binding proteins, as everybody knows, and at least one response is activation of an NADH oxidase, which then generates ROS, reactive oxygen species, and another signaling pathway is via MEP kinase to alter gene expression. So if you look at this scenario, and I think all described systems, it doesn't matter animals or plants or yeast or so, these are all phosphorylation events. And these phosphorylation events occur fast. So our goal was, we of course looked whether there are changes in gene expression and so on. We identified a number of genes which were differentially expressed and require a receptor for cellular oligomer-induced gene expression. But what was more interesting for us to see is what are fast responses. So do we see phosphorylation events? And what we did is we identified proteins which are phosphorylated or dephosphorylated within latest after 5 minutes. This was a bigger task. The computer was running 4.5 months, so it was not an easy experimental setup, a lot of probes. And one of the major protein that we identified that is rapidly phosphorylated actually within seconds is a protein that is necessary for cellulose biosynthesis. This is a cartoon showing how cellulose is synthesized. The final step of cellulose synthesis, this is a microtubule, and the synthesis of a cellulose starts within here. There is a cellulose synthase complex, which is then shifted to the plasma membrane, and a crucial player for that is this the cellulose synthase interacting protein 1, which is rapidly phosphorylated, and with this protein directs this complex at the site where cellulose is required. That means where the cell wall must be repaired. We can go a little bit further, and we did this extensively, and all the proteins in blue here are significantly phosphorylated within 1 minute after application of cellotriose. Cellulose biosynthesis starts in the ER here with all the precursors. I don't want to go through the details. Then the proteins are then shifted to trans-Golgi network and then further assembled here, and then it's shifted to the plasma membrane, and then ultimately this complex is also degraded by endocytosis, and the units which are then generated are then regenerated via exocytosis to generate new complexes. So all these proteins are rapidly phosphorylated, indicating that one of the first steps after breakdown of cellulose is activation of processes which lead to cellulose repair or cell wall repair. There are a number of other processes which are also quite important in this context. There are defense proteins which are activated. I don't want to go into detail, but I would like to highlight one, MEP kinase, which I think is very important in the whole scenario. This is MEP kinase 8. Not many people have looked at MEP kinase 8, but this is a candidate, this is a protein rapidly phosphorylated, and we are working on this protein right now. Again, an Indian PhD student. And we believe that this is a very crucial player to balance growth and defense. It's important to know that the cell doesn't know why the cell wall is degraded. So there can be a pathogen, there can be a friendly microorganism, there can be internal signals which stimulate or which degrade the cell wall because the cell must grow and then there must be a rearrangement of the monomers. So there can be many reasons why the cell wall is degraded. That means in order to establish a proper response, the cell must know why the cell wall is degraded. Yeah? And there must be other signals which are combined with the information that in the apoplast there's an accumulation of cell wall breakdown products. And there are a number of other signals, I don't want to go into detail now. And this response, ultimately the response that is established is then in balance between growth stimulation, for instance repair of the cell wall, or growing of the cell, or differentiation of the cell, or activating defense responses against pathogens, for instance. And it's always an important decision of a plant. Do I invest in growth or do I invest in defense? And a crucial player appears to be MAP kinase 8, because MAP kinase 8 is reversibly phosphorylated, very fast, very simple, and it, for instance, stimulates ROS production. I'll show you only one, or I describe you only one example. So if there is not enough reactive or reactive oxygen species, MAP kinase 8 stimulates transcription, transcription of ARBO-AD genes, phosphorylation of ARBO-AD genes, and therefore higher production of ROS. If it's too much, then it's all shut down by MAP kinase 8. The same holds true for defense gene activation. That means MAP kinase 8 directly controls phosphorylation of MAP kinase 3 and 6. So we believe that MAP kinase 8 is a crucial player balancing the response between growth and defense. And this is also a target of the signal from the cellotrios. Let me come briefly to, if I have a few more minutes, I would like to tell you a little bit about the malectin domain. Malectins were originally identified in mammalian cells in the endoplasmatic reticulum. They repair or degrade misfolded proteins, and during evolution apparently it became a crucial epitope in the apoplast of the plant cell. If we follow the secretion process, one can clearly see that this epitope in the malectin domain receptor kinase can easily become an epitope that is now located in the apoplast of a plant cell. And we found out that there are two conserved phenylalanine residues in all conserved malectin-containing proteins in Arabidopsis and plants. And when we mutate these two phenylalanines, the activity of the calcium, for calcium induction, we measured this as a level of calcium induction, is completely gone. So this is the wild type, and this is happening if we kill both phenylalanines or replace both phenylalanines. So we have some idea about the structural information. And we wanted to know whether other sugars can bind, and what is the difference between the animal system, the animal malectin and the plant malectin. So the quintessence of this is just summarized here in this cartoon. In animals, the malectin, got its name from maltose binding, binds almost exclusively maltose, a little bit of nigerose, but almost exclusively maltose, which is a dimer and has an alpha bond. If we go to higher plants, we get activation by cellobiose or longer. The cellotrios is the most active one, cellobiose is also active, and that has a beta bond. And it also can be activated by lactose, and to a lower extent by trehalose, but not by monomers, and not, for instance, and none of them is activated by saccharose. So what we can conclude, at least from the different analysis of the sugars which can activate the plant and animal malactin proteins, there must be a shift from an alpha bond to a beta bond in the recognition. And this makes sense because all the plant cell wall polymers are beta bound, have beta bond sugar moieties, and therefore we can speculate that this shift must have occurred during the evolution from an animal protein to a plant protein. Yeah, I think I will stop here and will briefly summarize what I told you. The plant cell wall is of course often degraded and must be degraded, must be remodeled. This can happen from internal signals which stimulate growth and development, but of course also external signals. The external signals can be, for instance, beneficial, but can also be pathogenic microorganisms. So there must be some sensory system, so the plant cell must know what is happening in the apoplast, that the cell wall is degraded. And the easiest way for the cell is to detect the breakdown products. In case of cellulose, these are cellooligomers. The most effective one is the cellotriose that is perceived by this receptor kinase, which I have described to you today. For sure, this is not enough for the cell to establish a proper response. There must be additional information which specify the reason why cell wall is degraded, and the combination of this information within the cell establish then a proper response. For instance, let's grow, let's activate defense mechanisms, and so on. And we believe that a number of crosstalks and a number of signaling compounds are important for synchronizing the response. For instance, the MAP kinase 8 can balance growth and defense responses. Yeah, with this I think I will stop now. This gives you just an overview on recent activities in our institute, which was mainly started originally by 2 PhD students working on Piriformospora indica, which was also coming from India. Ajit Verma brought it to us. And then some other PhD students worked on it, and it's now continued also with 2 Indian PhD students. And they sent me an email this morning. They had great results, but I shouldn't talk about them yet. Thank you very much for your attention, and yeah, there will be no questions.
Thank you very much, Ralph. It was a very exciting talk, and at least I'm talking finally in English, because when I was in Germany, I was listening all the time talks in German. I was feeling sleepy, but now I'm feeling very active. Thank you.
Okay, yeah. Not sleepy, that's good.
That's good. Then I'm requesting Dr. Sunti to deliver the prize to you.
Thank you. Thank you. That's nicely to your story. That's great. Yeah, yeah. Thank you very much. Thank you very much.
Okay, guys, I also want to add that I'm meeting Ralph after 33 years. Before that, we were friends in California because he was in Stanford, I was in UC Irvine, so we used to meet occasionally. But last 2 days ago night, we met after 33 years. It was amazing. Then he was telling to me I became a little bit taller. Then I said, no, because you are aging, you get a little bit shorter. That is why it looks like developmental balance. Sorry, I'm talking biology because I'm no longer plant scientist. So now I want to talk briefly about some introduction of my thing. First of all, I'm very thankful to the opportunity. Dr. Sunti, when I was walking to him the other time, she was telling everything is circumstantial or by chance or something. It may be true, because when I joined SLS, I was put under a different faculty member, but my seniors told me, if you work with that faculty, you will not publish anything after 5 years also. Then I got very scared, you know. Then they also told me that you can go to Sipra Mukherjee Sudhir Sapori Labs, that is very, very good, you will publish properly. Then I went to Sivatosh Mukherjee, he was a dean, he said, sir, I want to change it. Then I am sorry, there is no space, you cannot change it. Then I told him, what you will do if I don't change? I will leave this place, I will go to another place. I told him, he was really staring at me, nobody, then he came, walking from his seat to my thing, and he put his hands on my shoulder. I thought he was going to hit me or something. Then he said, Raj, nobody ever talked to me like this, and you talk so daringly, I'm really impressed, let us go and talk to Sipra or something. We came down step by step up to the third floor to second floor. I still remember, because when you pushed me, I don't think. Because the first time, it's also coming from south to north, all these things, but it was so nice. And then I went and talked to Dr. Sapuri, and Dr. Sipra was there. Sipra said, why not we take him to our lab? Then Dr. Sapuri told, sorry, I just now accepted this guy today morning. So then Sipra said, somehow we have to do something. Then Dr. Sapuri said, maybe we think about it, we can do something. But incidentally, the guy whom Dr. Sapuri accepted, he is my actually very great friend of mine. By first flute teacher that we became so close friends. Squash Koski, he was supposed to come, but he could not come. But we are so, I have also seen him after so many years. But it is a life it makes, like interaction. So that's why I'm very happy to come to this lab. But then, I want to tell you briefly about, there is my talk, guys. Yeah, yes, sir. Okay, yes, I have to briefly tell. So, anyway, I have to, I'm like we tell the circumstances, I'm also very fortunate to be coming to Dr. Like any other. So anyway, I have to continue here a little bit. So the important thing is that, like any other person at the time in JNU, they apply immediately to United Kingdom or USA. But I also started applying to United States to go as a postdoc. But because of Dr. Sapuri only, I went to Germany. Because that is also some circumstantial. Maybe that is how because he not only made my life, academic life, he also made my family life. Only because of Dr. Sapuri, I went to Germany. That is how I met my wife in Germany. So I don't know what never happened. My father is never to be on me. My wife says, what are you to come to Germany first? It's really funny, you know. It's very nice. It's very interesting. My wife also admires Dr. Sapuri. Even recently we greeted him from US on New Year's Day. Immediately she says, such a humble person, you are so lucky to have as your boss. You know what I mean? Even though she is totally different. Most importantly, he told me at that time, you can always go to America, but you will never get a chance to go to Huntsmore. There is also another Akira from Japan was also here. It is actually absolutely dream for anybody who is working on phytochrome to be in the lab, interacting with Hans Moore and Masaki Furuya. And one time I was they were all discussing in the podium. I also went talking, then somebody made a picture of that. I thought this is a God's blessing, to be in this, all because of Dr. Support. Otherwise, I would have not accomplished that. So, anyway, but I wanted to tell, if anybody wants to know why I changed the cancer, you can talk to me. But Dr. Sapore taught me, always think something which other people are not thinking. Look everybody what other people are looking, but think something different. This exactly he was telling to Ramaswamy also, because they were working on black salads. So that is how when I changed to the cancer, one of the famous surgeon asked me, Raj, I know you have a lot of plant signaling background. I wanted to, can you please help me about how to delineate cancer problem? Because he has two genes, RAS gene, he put it into the mouse brain cells, so then you will get a simple proliferation. You put AKT, nothing will happen. But when you put RAS and AKT, it becomes glioblastoma multiforme. So that is a terminator disease, means there is no medicine for that. People will die. So then he gave me that project, so I can work with him so that I have a lot of money to work on the cancer field. But it is interesting, later on I can tell why I went there, but it's not important at this time. So when I systematically again followed Dr. Sapore's suggestions to do experiments systematically, every experiment planned systematically like that. So I delineated every step, because every various possible signalings between downstream of RAS, downstream of AKT, and I came up with, you can see the RAS signaling as activating the translation initiation pathway, and AKT signaling is also initiating the translational ability pathway. Of course, this is I made it up because it is also there. The signaling pathways are there, but they also a plethora of signaling goes on underneath the RAS, underneath the AKT. But if you go all of those things getting confused, people are going and measuring the gene expression, At this time, in the whole world, in the cancer biology, people spent millions of dollars identifying this gene, that gene, publishing and overexpressing that gene in the mouse model. Oh, cancer comes. But I found something when they put together translation initiation of oncogenic transcription factors are only initiated, not anything else. So, but till I found, it is my fortunate or blessings of my supervisor, unless this is nobody has shown in cancer biology. 2003, my paper is the first time shows this, but incidentally, I submitted this paper to the world leader in the translation field before submitting my paper. So he was really shocked, what this guy is sending me such type of data to me to evaluate. No, they could scoop me in 2 minutes. And my real surgeon boss, he was very mad. Why you sent to him, he may be scooping you. But I had a confidence in him. But anyway, we submitted this paper, and I don't want to tell much about it. From 2003 onwards, we had a series of every month top journal is writing 1.5 page about my work. But as late as till last August, my paper is continuously called as a milestone finding in cancer biology. So you people have to little bit read when you have time. Sometimes there are very good prefaces on my work that will tell you this gave me extraordinary energy to work on cancer. So later on, at this time, then we also followed up this one with other collaborators. It is much later. Other collaborators in stone catering only. It is not only in brain cancer, even in breast cancer, transcriptional activators of oncogenic transcription factors are the first activated even for metastasis. So therefore the foundation of the grounds are there. But I told you the Nobel Prize winning candidate in Canada who received my paper first, he was the first person to show ER4E is the oncogenic factor. But I have shown, demonstrated clearly, cancer comes not by new gene expression, like millions of people work on it, translation initiation of certain genes. But today, John Rupert, somebody who was mentioning a paper, so she has lot of data, so many signalings. At the bottom there is a translation and transcription things were there. I was thinking how they are missing this, if you go to that, many of the genes may not be relevant, you may show overlapping, but you have to identify, you have to know what made these genes express. As you all know, transcription is nothing. Transcription can have a representation of stable messenger RNA versus unstable messenger RNAs. You may be carried away by taking hundreds of one. You can always, if you ask the bioinformaticist, you tell me what you want to show, we can analyze that way, we can show you that way. So that is how it can happen. So you have to be very careful whether it is functional or non-functional. So this is what only by blessing for me to have this. So then, similarly, at that time what happened is that I was immediately offered in Vistar Institute, director called me to my lab offering the job. that I could come and take up this on brain. I told I have no idea about cancer, I am coming from plants, this is the first paper I want to learn and I am scared to come and take up a job, I may fail, I may not get the tenure, So then I did not go. But then at the same time, urology chief walked into my lab and asked for my name, asked to see me who this guy is. But I was not there, so then he asked me to come to his office. I went there. Raj, can you please do me one favor, can you work on prostate? I asked him, what should I work? Whatever you worked on brain, can you work on prostate? That is what he told, simple. So, but anyway, I started delineating the signaling in the prostate cancer. So I found, my goal was to identify tumor initiating cells which are stem-like cancer cells. So I delineated again one more signaling very carefully, systematically. I found NF-κB signaling is very high in some of the cells that enhances the prostate cancer. But then I also found a specific marker, SOX9, which is a stem cell marker, which nobody has shown in cancer stem cells. For the first time I identified, fortunately, by again step-by-step delineating, systematically following the signaling cascades. So what happened is that when I found this in Fred Sart's lab in Montreal, Canada, he is a famous surgeon, prostate surgeon. He has systematically operated many thousands of patients, and he found 50% of the people whom he suggested margin negative. Margin negative means when tumor is taken out, the rest of the tissue should not be positive for any of the cancer cells. So he found all of them margin negative, but many people are, some 50% of the people are dying, 50% people are not dying. So he analyzed for various signaling molecules and he found NF-κB is positive. So what I found, NF-κB enriched molecules are stem-like cells. They don't proliferate significantly unless they turn on. So people who have NF-κB signaling, they are the people dying. That means they are left behind cancer stem cells. There is no marker to identify cancer stem cells at that time. So people say the margin negative, that means you still left cancer stem cells in the margin. So that is how they are getting the cancer. So this is another significant thing happened for me in cancer biology. Later on, collaborating with breast cancer doctors in Sloan Kettering, we also identified such type of stem cells exist in the bone. So 50% of not 50%, maximum metastasis from the breast cancer goes into the bone and stays there 30 to 40 to 60 years also sometimes. Maybe 20 years, very fast. So we checked this cancer-associated fibroblasts from the bone, and where there is no tumor cells, signals. But what we found is that when breast cancer metastasized to the bone, they receive in the bone cancer-associated fibroblasts. They will secrete certain vesicles, and these vesicles carry specific microRNAs, and these microRNAs go and turn on gene expression and make the normal stem cells in the breast tissue into the cancer stem cells. So we identified this also in experimental model system, but some using the PDX modeling of the breast cancer. In Sloan Kettering, extensively I developed PDX models of various cancers. It is a great opportunity for me because I was more excited to work with all other people in Sloan Kettering. Still works with cell lines. I tell them, you could do this work in Oklahoma or Kansas. Why you are in Sloan Kettering and doing this? You should work with my models. So I brainwashed quite a few people, and everybody uses my models. Not only there, Norway, Finland, Italy, and as well as Canada and San Francisco, UCSF. So I made significant markers, and I am recently offering to AMS. Some of the colleagues came, so they can also. I want to clearly tell one important point. Cancer is a systemic disease, and some people call cancer is not a single disease, multiple disease. I don't call cancer as a disease. I call cancer as a developmental defect. Like many people are having, we constantly have developmental defects. It is unfortunate certain genes will take awry and they become cancer. It is not cancer, it is not planned, and it is not coming like that. So this is an example I want to tell you why we have to study in a different angle. This is one of the PLX4032 for melanoma inhibition. The patient has a melanoma. So then you treat with this targeted therapy. Like you people go on signaling pathway, you identify one particular gene is there. If you express that, whatever phenotype comes. So the tumor decreases. But the same patient after 23 weeks later, for example, compared to 15 weeks therapy, then immediately develops much more aggressive cancers in the same place where the disease is cured, in the same place where disease was cured, and also in additional new places. So what does it mean? In the same place cancer did not really kill. You killed several other cells, but not the cancer stem cells. So because they will not be killed by the targeted therapeutics. So you need to target the cells that are really carcinogenic, So now I want to quickly go. This is also another model of medulloblastoma. So you treat the patient and the patient is completely cured. In the middle you have seen, let me go back here, in the middle is cured and again the disease comes back. So now I want to show what is the problem. The problem is targeted therapeutics. For example, this is the signaling axis. Forget about whatever we can know this upstream signaling, downstream signaling. So if upstream signaling results activation of these downstream signaling molecules, you may develop targeted therapy for this. Sorry, you may develop targeted therapies for this. But nature evolves such that when there is an obstacle to have a downstream signaling phosphorylation, they phosphorylate some other neighboring molecules. They will go and turn on the downstream signaling again, taking the rerouting of the traffic. You know what I mean? Something like that. So there are similarly, if you stop this signaling, they will activate some other signaling molecules downstream. These are all that. It is impossible to find out any human being with the available technology and methodology. So in addition, the human signaling map shows 300 different proteins, 900 signaling relationships. It's humanly possible, it's humanly impossible to identify cancer therapeutics. So the problem too is a developmental stage specific. If you take the colon cancer, I very often go to most of the operation theaters. The colon is taken out this much distance, for example this much distance. From here to here, you will have various signaling, various developmental stages. Normal epithelium, early adenoma, intermediate adenoma, late adenoma, and colorectal cancer metastatic stages. So if you take the Wnt signaling pathway is active in this stage, EGFR signaling, TGF-beta signaling, loss of degree function, and EMT and other signaling. So what you are going to treat the patient with the signaling targets, or with this, or all of them. You put all of them, the patient will die faster. That is it. So we cannot cure these things. That is why they cut it out. The patient lives a little bit longer. But if you want to cure, you need to know what is exactly the person, what is exactly the step we have to control. So in addition, nowadays, recently what is happening is that not only the tumor cells, which so far we believe, the neighboring cells and the immune system cells are also important. For example, targeted therapy, again I am telling you, estrogen receptor positive breast cancer cells can be targeted by estrogen receptor antagonists. So, but what happens is these blue ones will die, for example, and estrogen receptor positive cells can be killed, but the non-estrogen positive cells will turn into estrogen positive cells. So cancer therapy cannot be done not only by physicians, oncologists. Pathologists are the number one important people. They are the people who pull the trigger. Person will die or person will recover. The pathologists are wrong. Basically, oncologists will give a wrong medicine. They are not giving willfully wrong medicine. They rely on pathologists. Therefore, in Stone Catering, we have 21 pathologists for each patient. Each slide will run through 21 patients independently. So they will all tabulate and they will go for the treatment. Then, okay, for example, prostate cancer is a multifocal disease. When people develop symptoms with increasing age, we go to the prostate doctor, then they will take the needle biopsy. They will take 7 or 6, 7 needles. Needles go here, and needles go here, needles can miss this. So you take this, okay, we have this mutation, you have to treat it with this drug. But what about this? In the meantime, the patient will again develop disease progressing. So similarly, kidney cancer, 14 needle biopsies have 14 different genetic alterations. So again, each of them will be having totally different types of things. So how can you solve? Another problem of the cancer curing is this. So for example, carcinoma is an epithelial disease, sarcoma is a mesenchymal cancer, cell of origin. So if it is, there are some cancers, epithelial and mesenchymal cancers are together. So here again, the pathology is very, very critical. When you made the section, maybe the small post, this, for example, the whole thing may look like this, and this one people can ignore, for example. Not pathologists won't ignore, but I'm just telling. If they can ignore, when you kill all of these cells, these will proliferate very aggressively. So in my book, I have edited a book on cancer stem cells. I hypothesized, so far nobody disproved this, but nobody accepted it. What is my hypothesis is this. When, for example, let us talk in a normal layman terms. There is one, let us call, I don't know how, in the street. So street, there are some bad people are there, and there are two different gangs, one smaller gang and the bigger gang. I'm comparing because cancer is aggressive disease to these people. Smaller, so long bigger gangs are there, smaller gangs will not work, will not take over the normal people. When the bigger gangs or all police cops are arresting them, or maybe they put them into the jail or something, the smaller gang people will become active. Same thing here. So if you kill the aggressive cells, these smaller cells, they proliferate much more. So far, while the bigger cells, the aggressive cancer cells will try to constantly prevent the slowly growing cancer cells. So Rolf was presenting recently, just now he was telling about what makes even the cell is disrupted, cell wall is disrupted. But at least in cell membrane is disrupted, various signaling pathways are altered also similarly, and they are doing. The problem 5. So problem 5 is that this is the breast cancer model I developed. For example, this is the immunocompromised mice. Human tumor can be OR, you can bring operation theater, you can transplant directly into the mammary breast. So then the tumor grows and the tumor metastasizes to the bones. This happens in the patients. It also happened in the mouse models which we develop. But if you inject, taking this advantage, we can develop models which is for example, this is the mammary fat pad of the breast. We put it in the mouse and they metastasize to the lungs after the bones. For example, if you inject into the bones, they went into the liver. So it is also unluckiness. A person who has breast cancer, where it metastasizes to the lungs or to the bones, they can develop this type of diseases. So then importantly, the drugs developed for this disease will not cure this or this. That is why patients dying after metastasis. Primary cancer is cured. But I personally, I am not an oncologist. The oncologists say if the mastectomy is over for the breast cancer, fine. So we should be able to think about it. All these things, forget about it, and the last 2 slides. So that is why we are trying to take the T cells and engineer the T cells to express specific proteins. If we can identify on stem-like cells or tumor-initiating cells, we can target only those cells. Therefore, the cancer can be perhaps eliminated. I just want to show here. This is the CAR T cell approach we developed. This is all the tumors we started with is like this. Then we treated with the specifically engineered T cells, that is called CAR T cells. Our institution is one of the 2 in the world who developed CAR T cells. So I collaborate with them. They prepare for me whenever I need it. So we treat them. So we completely eliminate the cancer. So I want to tell, this is for example, I use kidney cancer. This kidney cancer specifically metastasized to the brain. I had a lot of interest in this one because Dr. Supra Mukherjee died of the kidney cancer went into the brain. So I specifically requested, I have an IRB, any cancer material, any patient identified operating sound gathering, I can have access to the specimen. So this specimen we generated, our CAR T cells, this is the tumor is growing, we treat, the tumor decreases, and if you don't treat, they have lung metastasis, and if you treat with the CAR T cells where that kills the tumor cells, the metastasis is decreasing. So these are all the preliminary approach. So this is the MRI of the mouse, the lung metastasis, and tumor is decreased with the treatment. So now all these things are done with the mouse models, but still we want to be very careful. We want to use the humanized signaling. For that reason, we are taking the mouse, immunocompromised mouse, we are humanizing the mouse. That is commercially available. I did some experiments, I was not successful. But I have got IRB approval along with one UK doctor joined in our institution. So we are making the patient specifically humanizing. That means the patient walks in with breast cancer. We take the CD34 positive cells, we inject those CD34 positive cells into the mouse. It expresses the mouse, that particular patient signaling. Then we know if the patient is going to getting operation, at least 1 week before we give this. So then we can treat. So this is the stage we are in. And in addition, we are doing the same experiments in a very sophisticated way. Nature, in fact, we explained to the Nature, along with the Cornell collaboration, Nature wanted us to submit. In fact, they are calling every 3rd week, is your paper ready? Is your paper ready? We want to look at it. But the paper is this. We took my model, breast cancer model I developed, primary cells from the patient, and Cornell breast reduction people are coming. So we take the tissue from that. We isolated stroma of the normal patient and the breast tumor cells, we mix it together. Then we are founding the macrophages, which are normally killing the tumor cells. They are bribed by the tumor epithelial signaling interactions. So they become, instead of killing the tumor cells, they are facilitating the tumor growth. This is how the tumor escape is happening. The treatment escape is happening. That is what we are working on it. We got very interesting results. We are doing that to another step layer, the state-of-the-art model, where we spots, like a spatial transcriptomics type of thing we do. That between a stromal cell next to it, breast cancer cell, and what is the signaling before activation and after activation, we can do. Thousands of messenger RNAs can be measured. So we are in that type of level, so I am very proud. Maybe one day we will have very good results to cure the patient, That is what I want to tell. This is all because of Dr. Sapore's advice. Instead of going to Davis and I become something else, I went to Germany and all these things, it happened. So that's what I want to conclude. These are my last things. Heartiest happy birthday, dear Dr. Sapori, Padmasri Sudhir Sapori. Sir, we all in our family express our sincere greetings and best wishes for many returns of the day, and also share the pleasure of this exceptional day of all of your family, students, friends and colleagues. May God bless you with the sound health, great energy and amazing guidance to many more people like all of us to date, along with the continued success in your love and dedication for science and sharing. Sorry about that.
So, thank you, Dr. Raj, for telling us, explaining your cancer research in a very layman language and telling how plant biology has helped reaching milestones in the cancer biology. So we take pride in that, and thank you, sir, for giving Raj Shekhar to cancer biology. So with this, we come to the closing of session 3, and also which we begin with the next session, and I'll ask again Dr. Raj Shekhar and Professor Hasnain to kindly moderate this session, and we move forward.
I'm thankful to Dr. Hasnan Etisham, so he immediately accepted our request, but I was very nervous whether he's so busy person, he will not come, but Dr. Sapori means he came. That is very nice of him. So Etisham and me, we are making this very small thing. We are inviting all the endowment committee members, which we on our own decided about 1 year ago to establish this, and we still have not lodged the money. We are still thinking where our endowment fund will be there, who will support us continuously without any obstacles every year to celebrate this. I wanted you people to understand, every year we will celebrate this, and Dr. Sapori's interest people, we are going to award the people who have worked on Dr. Sapori's interest. Those people will be rewarded with the money we have, and we will have small informal, but maybe every 5 years we will have such type of grand ones. Okay? So we request all the endowment committee members to come here and stand by, so just for a photograph or something like that. So I want to thank Neeti Mishra and Charan Preet. These people, I am really bothering them like crazy in the middle of the nights and everything for me, not for them, because I come back and that is the time I have free time. So, and Arun Sharma and Girdhar Pandey and Raghuram, they were always available whenever I wanted, so they were coming to us, helping us. So we are just showing, these are the endowment committee members. If you have any ideas to do better than what we have done so far, we will look forward to that. And we also want to thank Meena Sapori and Sailaja Sapori for their kind support for that. They did significantly their level best. And Etisham promised me, come on, sorry, why we are putting Rolf is not because of he is my friend, he got the award, the endowment award, so therefore we are putting this. Hasnan, you also come. Yes.
So it's setting a very high bar with the first award, and
Yes.
if we don't maintain this bar, whom to hand?
If anybody wants to, please come. Yes.
It's okay.
Yeah, maybe I should wish him a very happy birthday, because since morning I forgot and I was so busy not. Yes, and he has always been an inspiration for not only me but for all of us, and we have heard that throughout the day. And from my side, if you would ask me, being a plant person, I can just describe him as a huge fruit-bearing tree, because once you go under that tree, you get not only thandi hawa and fruits and everything, oxygen, and that is what we get. And even if somebody throws a stone at it, there is only a fruit or a good thing that is going to drop down. So this is how I look at it. And I know, because I've had rough times. Science, we have up and down. And during one of the rough phases, I went to sir and he says, look at a seed. You just throw the seed somewhere and it will grow. It will keep growing silently. Sometimes the growth is slow, sometimes the growth is fast, but the seed keeps growing silently. So this was a very, very important take-home message. He said, keep growing wherever you are, whether you grow slow or whether you grow fast, but keep growing. And this is a very important lesson that I've learned from you, sir. And I think I'm still learning. I still have a lot of learning to do. And so wish you all the best. And hopefully we can all, you continue to inspire all of us, and we continue to learn from you. Thank you.
I think all of us have been greatly benefited, whosoever has been associated with sir, and he's still very active. We can't think of being active till that age, the way it looks like, but he's still going strong, and I wish that he has many more years of active research and good health and happiness. Thank you.
Yes. So one thing we learned from Professor Sapori is never laugh at a student's crazy idea. Never discourage him. Tolerate all his idiosyncrasies. When I handle my students, I know what that meant. It took quite some time to get into those shoes and learn that same level of tolerance that is becoming increasingly rare. Everybody thinks it's their right to run their lab the way they like. But students have their right to education too, and they have their right to mistakes too, and they have their right to learn from their mistakes. So we need to develop that large heart, and if not for Professor Sapori's large heart, people like me would never have had a chance anywhere else. Thanks to JNU, thanks to Professor Sapori, and thanks to all the learning all of us do together. Happy birthday again.
Thank you, sir. I think everything you have done for us, I think this is the time we should be very thankful to you, and I just want to express best wishes to you, good health, and hope we continuously and every year we celebrate this event with a lot of joy and charm. Thank you very much, sir.
So after now we are going to watch 30 minutes or so of the comments. Yeah.
Okay, so see, this was one event where people who are presently not in India or not in Delhi for the moment also wanted to join in. So and it was not possible for everybody to come here, so we have taken some video recordings of that. For the next 20 minutes or so, we'll play that recording, because anyway these people we presume are joined to us online with the link that is going on.
So first we'll watch 30 minutes. That is one. But after Dr. Sapori speaks, again we will have some more total video time is 1 hour 14 minutes, but after 30 minutes we are having a break. Dr. Sapori will talk, and we will go for coffee. If you want, you can come back.
Yeah. I just thought that some of us in the audience may want to say a few words about his or her association. So our only request is do not exceed more than 5 minutes. I also see Professor Ajit Verma here. So I would like to invite Professor Verma also to come and say a few words about his former colleague. Let me start with Professor Ajit Verma first. I see him sitting here. And then anybody can please feel free to come. Just take a few minutes.
Thank you. Myself and Sudhir Sapori came to know each other in 1975 when I joined GN, the School of Life Sciences. And we have been a great friend since then. I know that we both have been Humboldt Fellows. We both have worked with Rolf Muller, wonderful scientist. And I wish Sudhir a long, happy life, and also the Meena and the family children. Thank you, Sudhir. We wish you a long, happy and healthy life. Thank you, Sudhir.
Thank you, Dr. Ajit Verma, for stopping by. It's very nice of you. Yeah, thanks.
Can we raise hand those who would like to say a few words? You don't have. Please. If you can come here, fine. If you can't come here, we can speak from there. But don't exceed 5 minutes.
Yeah, actually, I don't know. I was just looking through my old papers and I previously met him, sir. Well, way back in December, on 20th December 95. So I'll just read out what I don't know what had happened in the lab or what made me to write. A colossus in a cog, seen always in a slog, found him placid, thin and frail, always ever ready like a rail, with nimble and jovial nature, thus makes him a rare creature, a hidden treasure of knowledge which masses acknowledge, an enchanting personality with a strong instinct of originality, masses vouch for his sincerity, who is always in search of integrity, a committed teacher and not a preacher, thorough researcher but behaves like a fresher, an intoxicating speaker which fascinates every listener. His presence calms a thunderous roar which never makes one otherwise bored, mostly right, rarely wrong, but always found him very strong. His single encounter leaves you to ponder about this little master's wonder, where willingly or unwillingly many surrender. Yeah, it is none other than my respected teacher, Professor Sopori. Thank you so much, sir, for being there for me many times when I had problems, personal health problems or research-related problems. Sir has always come forward, given very valuable suggestions. And when I moved to DPT, whenever somebody will tell me, with whom you are working, I would say, Professor Sopori, oh, such a nice man, but very hard taskmaster. So those two things would always go simultaneously with whom I have met. But sir, even today, whenever we need sir's advice, sir is always there for us. Thank you so much, sir, and wishing you a very happy birthday. Thank you so much.
I see any. There's a. Yes, please. Another colleague of Professor Sopori. You can come here. I know personally. I thought let him introduce. Known him for a long time. I didn't want to introduce you because I want all the best. I want you to introduce yourself.
He has said, Hasnan, many of you may not be knowing. I am Biren Malik, and I know Professor Sopori since 1986. And Vaishnav is here, of course. Again, he is senior, but we are the same batch of selection at JNU Life Sciences. And about Professor Sopori, many things must have been said. He's a nice person, nice scientist. Namaskar, Bhaviji. So, Neera is also here. So Neera, Vaishnav, all are much senior to me, but we are the second generation of selection of life sciences. All selected around 95, 86 and joined there at that time. They are from the plant sciences. My subject is from neurosciences, but then why it is important to say, why I like about university setup, and with Professor Sopori, I still relish the day, and Niti is here. Professor Sopori and me were talking one day about it while taking tea, the plants sense light and then flower. And that generated how the electrical signaling moves in the plant from root to shoot. Niti was those days doing PhD, and I remember late evening from 5:36 till late 8:30 to doing the electrical signaling part, and that is how it came forward. And Professor Sopori was very kind to even give me an authorship, which probably was not that important because I was a technical person to do that, right? Otherwise, very nice, fantastic food from Bhaviji. And then coming from academics to administration, he was the vice chancellor. And then I happened to be dean those days. Of course, one thing is very difficult to get something which he thinks is not to be done. But the positive thing is, if he thinks he is convinced, it will be done. That is the beauty of the administration, and more importantly, the academic administrator. And that is how he could understand, even we could do that, the stabilizer system, and from whatever it is, that is still life sciences going on, we got the third thing. That is the academic administration. And then in the family, the great thing with his aunt and Bhabiji looking after there, and raising their two kids so nicely. And many people must have gone through the lab and whatever might have happened. I might not have been directly involved in his lab, but I got the flair of the science. It's a pleasure to talk to him on many, many issues. So with this, I wish him all the best, happy birthday, and to Bhabiji and to their children, to all the best, to have a nice family and growing further. And if there is anything, we would like to have more discussion and your lecture sometime. I'll talk to you on this issue later. Thank you again.
Because I may not get a chance when I have to say 15 minutes something about Biren, because sometimes you forget many names, things. You know, in fact, one day I was in my room in life sciences, and Biren walks in, and he found that I was sweating, and there were some issues. He said, "Are you okay?" I said, "I'm okay, but there is slightly some pain and other things." He's a doctor also, a different kind of doctor. He said, "I feel there is something wrong, and there must be some heart problem in there." So he immediately took me home, took to Meena. They said we should shift him immediately to the hospital. He took me to the Sabdajang hospital, he was carried. So I'm alive today. Thank you, Biren, for that.
Thank you. Quickly, 2 minutes.
Well, I cannot pass this opportunity to speak a few words about Professor Puri. When I joined Jawaharlal Nehru University as a student in 1976, Professor Sapuri interviewed me as a student to be a teacher, to a student in the school. From that day onwards, when I joined, and he has been a great guiding force to all the students at School of Life Sciences. When I came to JNU, I had a very big interest of working on photosynthesis. So I just came to JNU to work on photosynthesis, and I joined Professor Mahanti those days. And but the phytochrome was another interesting photobiological subject to me. So I was really deeply connected with Professor Sapuri and also his students those days, and my friends those days, R.P. Sharma, who is present here, and Sihag, who is also abroad, and also many other, Mohan Jain, Neera, and many other colleagues who are working with me. So I came to know much more about phytochromes after coming to JNU. And of course UC Vishal was also there. So all these people were really a great opportunity for all of us. Of course later on, other people joined, and even who else joined? There are many other groups who joined. Sangeeta joined, and many other people also joined in this group. So it was a really interesting time for me in JNU when I was a student, and also when I came back and joined as a teacher in JNU in 1987, then also I knew him very well. Of course, he knew him very well before, and we also collaborated on many issues, in scientific issues. And it was also my great opportunity at some certain time in my life to organize an international conference in his honor on his 60th birthday. I feel that it is a great honor for me to do that. And then, of course, I went out, he went out of JNU, came to ICGEB, and went back to JNU. and and then he appointed me as the Dean of School of Life Sciences, and he was my boss also. So he was my teacher, he was my colleague, he was my boss. So with these words, I must say that Professor Sapori is a great mentor, is a great teacher, and a great scientist. Thank you, sir, to be always with us. Thank you very much.
So now we'll play some videos, we'll take a pause, and then we'll pause the videos in between, and in the meantime we can line up a few speakers.
Happy birthday to you. Happy birthday to you. Happy birthday. Madam sir, happy birthday sir.
Let's go back to your childhood.
I had 5 brothers, 6 brothers, we are there, 1 sister.
Happy birthday to a wonderful man. I'm so blessed to have you in my life, as you have brought so much of joy and happiness, not only in our family, but others too, as you are the most patient, helpful, honest, and a lovable person. You always deserve the best. May God bless you and keep you in excellent health.
Of your family sometimes. Because my wife, who has been a great support to me all these years, has never complained. Even at home, I have my time schedule. I come to my office here at 8:30, whether I'm at home or not. I will go for lunch, have half an hour break, come back, and stay here till I finish my job. So it's a regular routine. Whether I'm at home, at my office, the routine does not change. And I'm so lucky that I found her, that she has all these 50 years never complained once that why there is no time for family.
That's why you decided to join science? Or was it influence of parents or teachers that motivated you to do science?
Right till the end of schooling, there was nothing very specific preference for science or any other subjects. And I had also thought of getting into law at one time, and or had some liking for and also
good scientist.
And it was during that honors course I got more exposed to the life of the plants, and that stuck with me at that moment. And when I went to master's program, I took botany as my master's program. I think in my master's program, one of the teacher, Professor Kaur, he was a great teacher. He was teaching us lower plants and fungal systems, how these grow, their life cycles, and also the genetics background based on which these plants or these lower animal plants respond to different environmental conditions. He was a great teacher. Even today, when I meet him, I talk to him, he's still in Jammu, and meet him, I think there is always something to learn from him. more focused on that I should get into either physiology, genetics areas. And then I came to Delhi. Advertisement from Delhi University, Professor Satish Maheshwari, who is son of the famous Panchanan Maheshwari, the great botanist. He himself had done a lot of great work. So I went to him and said that I'm interested in doing my PhD in Delhi University with you. He interviewed and said that you have to work with me for 2-3 months without fellowship. If I find you suitable, then I will keep you. And then your fellowship will start. So let me see how motivated you are. So I continued, I was staying with my brother here and traveling in DTC buses.
Must be very hard for you.
From Lodi Road to the university. For 3 months I worked with him without anything. And then he finally, I know what he found. He said, no, I think you can be in science.
You have a potential to be a scientist.
He was another great person, who motivated me in doing science in a way where you can ask right questions, define what you want to do. And how to speak, how to write. I think he was a great teacher. I still admire him for whatever he has shown the.
Have a great day. Thank you so much. Love you.
Dearest Nadu, we all think you're so wonderful and amazing, and we all want to wish you a Happy New Year, Merry Christmas, and especially have a great, happy 75th birthday.
Happy birthday, dear Dadu. I love how you're so calm and collected. You inspire me to be just like that too. Have the happiest birthday wishes of them all.
Dear Nanu, wishing you a very happy 75th birthday. We will forever be grateful for having such an inspiring person in our lives. I admire your dedication and passion for science. You're one of the busiest people I know, and yet you take out time to talk to us about school, books, life and philosophy. You've been a part of so many of our fondest memories. So this is not just a birthday wish. It's also a thank you for buying us books, always encouraging us to be curious, and most importantly, for teaching us that there is always something new to explore. A very happy birthday, Nanu. Happy birthday, Nanu.
You've seen it growing, or you grew with the university, or university grew with you. You've seen good times, bad times, emergency, all those things happening here. Very vibrant public university. What has been your experience as Vice Chancellor?
This was a very, very different kind of experience for me. But being a Vice Chancellor, when you interacted with every faculty member in this university, that I made it a point that I visit every school and center. I talked to all the students and got feedback from them. I thought...
those were all always open for discussion?
Any student could come. We had open doors, students can come. But I used to usually discuss academics with them. Of course, for issues like hostels and others, there was separate timing for those.
चलो मैं कितना भी भेजी उसको समझ और सिर्फ समय देते थे, ऐसा नहीं था कि सिर्फ सुनते, उनकी समस्याओं को सुलझाते थे। और कई बार क्या होता है कि जैसे बैरियर हो जाता है, कई बार क्या होता है कि हम हायर अथॉरिटीज़ को खुद से अप्रोच नहीं करते। कोई ऐसा हायर अथॉरिटी मिले जो आपके हॉस्टल आ जाए, उसको ये फिक्र हो कि वो बच्चा कैसे रह रहा हो, अच्छे से ऑफिस से निकल करके हॉस्टल आ जाए, और बहुत बड़ी बात। तो ये कंपैशन उनका शो करता है। और ये मानना कि हर किसी का सोल जो है एक ही, जैसे होता है कि जैसे हम लोग साइंटिस्ट को मानते हैं कि वो मतलब कि आप स्पिरिचुअल नहीं हो, लेकिन एक साइंटिस्ट स्पिरिचुअल माइंडसेट का भी हो सकता है। मतलब उनका ये कहना था कि हर किसी का सोल तो एक ही है। ये बात सबके लिए केयरिंग, किसी भी बेसिस पे उन्होंने कभी किसी के साथ भेदभाव नहीं किया, कभी किसी के साथ दोष नहीं दी, किसी का लॉस नहीं हुआ। अच्छांश है, अच्छांश को सेरेब्रल पैल्सी है। जब वो पैदा हुए तो उनके ब्रेन फंक्शन कमी है, जिसकी वजह से भी मान लेंगे, और वो 95% डिसेबल्ड है, और वो क्वाड्रिप्लेजिक है। तो स्पास्टिसिटी की वजह से काफ़ी डिफिकल्टी उनकी लगा। तो सर जो है उनसे मिलने उनके हॉस्टल आ जाते हैं कि अच्छांश कैसे हो, क्या चल रहा है, कोई दिक्कत नहीं। तो आपका एक वाइस चांसलर आपके हॉस्टल आ जाए तो बहुत बड़ी बात है।
It is very fortunate that we have Dr. Akshans Gupta with us here in the auditorium, and I'm handing over the mic to him. So his special wishes, sir, on his birthday.
First of all, I wish a very happy birthday to my favorite vice chancellor. मैं उनसे पहली बार 2011 में मिला था, तो वो मुझे इतना ईज फील किए कि कभी भी लगा कि नहीं कि वो वाइस चांसलर और मैं एक सिंपल स्टूडेंट। उनसे मैं जब भी बात करने गया किसी भी प्रॉब्लम बता टेक्निकल चाहे पर्सनल चाहे एडमिनिस्ट्रेटिव, उन्होंने एक हमेशा पेरेंट की तरह मैंने सॉल्यूशन देने की कोशिश की। अब मुझे याद है कि जब मुझे मलेशिया जाना था प्रेजेंट करने के लिए, तो उन्होंने काफ़ी मेरी काफ़ी हेल्प की। जब मैंने 2015 में PhD कंप्लीट किया, तो उन्होंने मुझे में कोई कन्वोकेशन सेरेमनी नहीं होती थी। उन्होंने एक स्पेशल कन्वोकेशन को ऑर्गेनाइज किया। जिनों के जितने पब्लिक प्लेस हैं, चाहे ढाबा, चाहे होटल, चाहे लाइब्रेरी, चाहे हेल्थ सेंटर, सबको उन्होंने फ्रेंड बनाने के लिए अफॉर्मेटिव एक्शन लिए। अब मुझे याद है कि इन 2013 जब उत्तराखंड में कमेटी आई थी जून में, तो हमारे 3 फ्रेंड भी उत्तराखंड गए थे। उस दिन ऑफिस बंद था, तो हम 3-4 स्टूडेंट विजिटर के हाउस में, उन्होंने ना प्रॉपर हॉस्पिटल के बाकी एक इशू को सुना भी, और अपने पर्सनल लेवल से जितना बेस्ट हो सकता था तीन स्टूडेंट का लोकेशन फाइंड आउट करने की कोशिश की। इवन हर बार की खबर वो पर्सनल मोबाइल से किसी स्टूडेंट को इन्फॉर्म कर देते थे। उन्होंने 2011 से एक वीसी दरबार लगाना स्टार्ट किया जिससे जेएनयू से एफिलिएटेड हर बंदा, चाहे वो स्टूडेंट हो, फैकल्टी हो, नॉन टीचिंग स्टाफ हो, मिस वर्कर हो, हर बंदा वाइस चांसलर से डायरेक्ट बात करता था। I am very privileged that I studied in January during his tenure. Once again, I wish a very humble and happy birthday. Thank you.
We must really appreciate the efforts of Dr. Gupta to have come all over to ICGEB, and a very special greeting for sir.
Dr. Akshansh Gupta.
Happy birthday, supporters. May God bless you always with great health. I still miss you, sir. You were an able administrator, and times when I met you, your reflection helped me to understand life. Your empathy has kind of influenced me to do well. Sir, you are a remarkable person. I would want to follow your footprints as an academic, as a human being. Happy birthday, sir. God bless you always. Happy birthday.
इंडियन स्पाइन इंजरी सेंटर गए हम, वहाँ पे हम लोगों ने असेसमेंट करा दिया। वहाँ के जो फाउंडर थे वो सर वो पद्म भूषण अवार्ड और माउंटेनियर थे। नेहरू जी के समय में नेहरू जी ने इच्छा जताई कि बेटा मैं चाहता हूँ कि कोई माउंट एवरेस्ट झंडा फहराए, तो वो सर उन दिनों माउंट एवरेस्ट झंडा फहराया था 1965 से 65 या उससे पहले पड़ा नहीं, नेहरू जी के समय में था तो 65 से पहले ही था। मतलब वो पहले माउंटेनियर का जो ग्रुप गया था उसमें वो सर मेजर हरपाल सिंह हुआ गया सर, तो हम लोग गए। उसके बाद हमने सोचा कि क्यों ना ये बात जो है सर को बताई जाए, तो सर से हम लोगों ने कहा कि ऐसा ऐसा है। हम लोग वहाँ गए थे। जब अगली बार हम लोग ISIC विजिट करने गए कि हम लोग अपने वाइस चांसलर को यहाँ पर इन्वाइट करना चाहते हैं ताकि आप उनसे मिल सकें, तो सर हरपाल सिंह अजवाले सर ने बताया कि आपके वाइस चांसलर कल आए थे आपको बुलाने के बाद। यानी कि हम आज गए थे मिलने के लिए और नेक्स्ट डे हम VC सर को बुलाना चाहते थे, और हमारे वाइस चांसलर सर जो है एक दिन पहले आकर हम सर से मिला है, हमें बड़ा अच्छा लगा। और जो बच्ची है शिल्पा जिसका बताया मैंने हाथ वाला, वो मतलब उसको उसके प्रति बहुत ज़्यादा सेंसिटिव थी। मतलब जैसे होता है ना कि कुछ लोग जैसे सेंसिटिवनेस आप एक्सप्रेस करते थे बच्चे को बहुत दुख है या लेकिन उनका ये अप्रोच नहीं होता, पॉजिटिव तरीके से मिलते हैं, एज अ नॉर्मल ह्यूमन बीइंग ट्रीट करते थे। लेकिन उनका कंसर्न ये रहता था, उनका ये कहना रहता था कि जो डिफरेंटली एबल बच्चे हैं दे नीड एक्स्ट्रा लिटिल एक्स्ट्रा असिस्टेंस एंड लिटिल एक्स्ट्रा केयर। तो शिल्पा के लिए भी एक्सेप्टेबल थी। शिल्पा को उनसे मिलकर बहुत खुश होती थी कि अरे बात सर ने हमें इतना इंपॉर्टेंस दिया, मैंने कॉल घुमाया और सर आए। या फिर कई बार ऐसा होता था कि DC ऑफिस से या तो उनके पर्सनल सेक्रेटरी कॉल करते थे या सर ही खुद कॉल कर लेते थे कि आ जाओ, इसको दो बातें कहेंगे। कभी कभी कुछ बातें हमारी एक मीटिंग में पूरी नहीं होती थी, तो फिर होता था कि दोबारा आ जाए, तो हाँ दोबारा भी आ जाओ, दूसरी बार आ जाए, हाँ भाई दूसरी बार भी आ जाओ। तो जितनी बार हम लोगों ने चाहा उतनी बार। जब दोबारा से सपोरी सर ICGEB ज्वाइन करी तो एक दिन बैंक के काम से आया था, तो मुझे बैंक में मिल गया। तो मैंने मतलब कैच हो गया आशीर्वाद लगा। उसके बाद मैंने कहा कि सर एक बच्चा है, मतलब ऐसा उसका एक्सीडेंट केस है, मैं आपसे मिलाऊँ ना उसको? तो बोले हाँ हाँ मिलाऊँ। तो फिर वो ढाबे बैठा हुआ था, सर भी हमेशा ढाबे आराम से ढाबे बैठते हुआ काम सेकंड की सवाल कर ये बचपन में मैंने किताबें पढ़ी थी कि मतलब जो वाकई में अचीवर होते हैं, जो जिनके पास नॉलेज होता है, जो पूर्ण होते हैं नॉलेज, वो बिल्कुल मतलब फलदार वृक्ष की तरह झुक जाते हैं।
Okay, so we pause here, and if anybody else wishes to speak, yeah, we'll resume this video. So this will anyway be online, but for the moment we have paused. Yeah, please.
Happy birthday, sir. I have an indirect association with Professor Sapori. My wife was in his lab, worked for several years. But let me tell you one thing. I got my first job as an assistant professor in Delhi University Botany Department. Sir was there. I became a reader. He was again there. I was again a professor. Was there. So he was part of my career, and always like what I am today, and if I am here, I owe to Professor Sapori, because everybody needs a job. At the time of crisis, I got a job, and he stood by me. And then when I moved to Northeast to head the institute called Bioresource and Sustainable Development, he was also the chairman of the Scientific Advisory Committee. And when people don't usually dare to come to Manipur, sir, you remember when we had Dr. Sonti was also there. It was a very, very difficult situation, but he said, no, I'll come. And I remember that meeting for 2 days with 5 commandos. He was guarded to the hotel, and we couldn't move out of the hotel, and we finished our Scientific Advisory Committee meeting. But one thing is I liked about him, in spite of he being busy, whenever I wanted any help, he never said no. He said ki aa jao. Such a wonderful person. Me and my family, we are all benefited from his guidance. Sir, we are really grateful to you, and I am very happy to be part of this celebration today, and keep showering blessings and sons. Thank you.
दिल में एक ख्वाहिश जगती है कि मतलब अपनी पर्सनालिटी भी हो तो वैसी ही हो।
On the thing I was doing. So ma'am, I wanted to ask sir, but I'm feeling nervous. How should I talk to sir about my doubts? Nervous होने की बात नहीं, मुझे तो वही लोग सर से नहीं करनी। Yes, he is very good, you know. मेरी बात नहीं है सर, he is very अपने मन। Okay, यार ये कोई बात नहीं हुई। In fact, मैं भी जब first time में से मिली थी तो मुझे बहुत ज़्यादा hesitation था, लेकिन सर तो बहुत ही soft spoken है और सर ने बड़े आराम से पूरी बात करने की बात है।
Mom, you have done PhD at sir, and you all people are associated with him since long time. And if we want to define him by his name initials, how can we?
Do you mean like from each letter?
Yes,
there's some
yeah,
quality?
yeah.
Okay. I think S से तो simple first of all, and U से तो understanding, personality और thing.
D से I think dynamic leader. X से humble.
I said I think sir is the most inspiring person.
And medium too.
विदिशा सबसे ज़्यादा एक्यूरेटली बता सकती है, मैं इसका रीसेंट एक्सपीरियंस है सर के साथ।
हाँ, मेरे पास जो इंस्टेंस है वो ये कि जब मैं सबसे पहले इंटरव्यू के लिए आई थी सर के पास, तो आई डिडंट नो सपोर्ट सर इतने बड़े साइंटिस्ट हैं, एंड then जब मैं उनसे मिली तो उन्होंने हमेशा इंकरेज किया है। एवरी डिस्कशन हैज़ बीन अ लर्निंग एक्सपीरियंस, हमेशा कुछ नया नया सीखने को मिलता है, कोई डायरेक्शन मिलती है, इंसाइट मिलता है, तो आपका काम आगे अच्छे से बढ़ता है। सिर्फ साइंटिफिकली नहीं, लाइफ में भी वो बहुत कुछ अच्छा सिखाता है।
सबको हमारे लिए बहुत अच्छे एक्सपीरियंस है सर के साथ। सर से तो मुझे बहुत कुछ सीखने को मिला है, वर्क एथिक्स एंड बीइंग पंक्चुअल ऑन टाइम, एंड मोस्टली सर हमेशा कुछ भी स्ट्रेसफुल सिचुएशन होते हैं उसमें इतना टैक्टफुली सर हैंडल करते हैं सिचुएशंस को, एंड ही नेवर लूजेस हिज पेशेंस। तो ये बहुत बड़ी क्वालिटी है। मुझे तुम भी कुछ बोलो।
सर हमेशा मतलब बहुत अच्छे से समझाते हैं, जब कुछ भी पूछो तो सर ऐसे बहुत कामली समझाते हैं, अगेन एंड अगेन, मतलब सर पेशेंटली समझाते रहते हैं अच्छे से,
डर नहीं लगता
और सर को गुस्सा भी नहीं आता।
हाँ, ये बहुत बात है। आप लोगों की बात सुन के मुझे काफ़ी इंस्पिरेशन मिला है सर से जाकर आराम से बात करने का, और जो भी कुछ डाउट्स हैं उनसे जाकर पूछना। थैंक यू सो मच।
I am on the comment with you.
So on this note, we would like to wish you a very happy birthday, sir.
Hi everybody, I am Dr. Abdar Matu from USA. I am a scientist with USDA at Beltsville, Maryland. I had the greatest pleasure of knowing Professor Sudhir Sopori since decades ago. He also worked in my lab and produced excellent research during that period. On his 75th birthday, I send him best wishes for good health, continued success in mentoring younger scientists, and long, happy life. So, Sudhir Sopori is a very gentleman type person who has been always helpful to scientists, younger scientists, senior scientists, and has contributed a wealth of information to the scientists in the United States as well as in his own country and other countries. I wish him really a very, very wonderful birthday celebration, and we hope he lives a long, very good life, and I congratulate all of you for organizing this thing. Thank you very much. Bye-bye.
I think time has come to invite Professor Sopori to say a few words. But before that, I would like just to take a couple of minutes to talk about my association with Professor Sapori. I think as you must have felt, I didn't have the fortune of working directly with him, but I was one of the first batch of students to have joined Jawaharlal Nehru University. We were, I think, a bunch of half a dozen students, or maybe 10 students, who were selected to start the program in India, first time in India, MSc Life Sciences program. This was way back in 1975. It was emergency time, and it was complete calm in JNU at that time. But 77, when we finished our masters, that is the time. But 75 also, we had such wonderful relationship with faculty there. As I said, it was very easy. We never felt that he is a professor or he is my teacher. We used to go out for, this was the kind of environment that JNU built. Subsequently, a lot of things had happened. I was very active. We heard a little while ago about, we were also quite active. Emergency was lifted in 77, and we decided to remove the guilty four. I don't know, those of us who are of that era will remember what was guilty four. The Vice Chancellor, Darshan Kumar, two other people, I don't know, maybe he may remember. But anyway, so that is the time when we took over the entire university. The university was declared sine die, closed, and Professor Sapori, we allowed some people to go and work in the lab, but we said we will not allow the school to function. We were teaching ourselves, Vaishnav, myself, and we were quite active. But the good part that I found was, those days it was very easy to talk to faculty as if we are friends. I remember Professor Shivatosh Mukherjee's example was given. He used to smoke very expensive cigarettes, and I used to smoke Charminar. That time, Charminar was the easiest and the cheapest cigarette available at that time. And even then, so quite often, Professor Shivatosh Mukherjee will offer me cigarette piyega. I said, why not? So he said, machis mere paas rakhte hain. हाँ, machis तो रखोगे तुम, cigarette नहीं रखोगे. So that was the culture of JNU at that time. Grew up in that culture. And subsequently, I think when we had the control of the university, few people were allowed to come and work in the lab. Professor Sapori was one of them. And I used to see him with a whole bunch of students, including Rajshekhar. I think Rajshekhar was also there. He says that he spent first few months in my lab when I was with Professor Kailash Upadhyaya. But it was fun time. It was great time. Subsequently, we left. I left JNU. I joined Delhi University. He was very happy that I have joined Delhi University as a full-fledged assistant lecturer. I was in the botany department. But I didn't like the culture in DU and culture in JNU. I decided to take leave, and when the DU was kind, they allowed me to go out for a postdoc. And then after that, they fired me. They said, you have to come back or we'll dismiss you. I said, dismiss me. And that was the time when I think we got back again. And after that, I had occasions to work for different institutions. And I must tell you one thing. Whichever institution I worked for, I took a lot of advantage. I'm very happy that Professor Sapori, immediately I will call him and said, I want you to be in my executive council. He said, Itisham, I'll be there. And he will be with me. And whatever we could do, whether it was Center for DNA Fingerprinting and Diagnostics, or University of Hyderabad, or Jamia Hamdard. Jamia Hamdard, I think you were also the member of Executive Council. He was a member of my academic council. It was a pleasure to, the kind of advice that he will give. Very honest advice. Ye mat karo. Don't do this. And that is something which I really liked. Honest advice and something telling upfront that this is not the way to go about. I really admire, and I'm very fortunate to Rajshekhar to allow me to come here. When he called me a few months ago that we are doing this special function and I want you to be there for this meeting, I said, okay, I'll definitely try to see that, and finally I'm here. I'm very happy. Thank you so much for inviting me. And the good thing that I learned was, I was not aware of the kind of diversity that Professor Sapori has been supervising over the last 50 years of his career. He's 75, so I think he must be having 50 plus years of his research. Amazing. And I find that so much of commonality between the plant system. In fact, plant systems are far more complex than the human system. And we working on humans and human pathogens think that we are dealing with very difficult things, but here it's far more difficult. And the kind of simplicity, the way the students are today, when I heard the presentations, it's all reflection of the boss. The boss is Mr. Sapuri. Thank you so much, sir. You have been a great source of inspiration, and may you continue to inspire all of us, those who worked with you or those who were not so fortunate to work directly with you. Thank you once again. I would now request you to say, talk to us about you. You have time with you. Till 6 o'clock you can be with us. There is absolutely no problem. But thank you once again.
Thank you, Atishan. I'm happy I'm not giving a scientific talk. Because giving a scientific talk in presence of your wife and daughter becomes very, very difficult. But what I'm going to talk to you about briefly, I don't think I want to because many things have been said. Firstly, thank you everybody who is in the hall, who have said so many things on recorded messages. And whatever is said, thank you so much for being with me here. And I thank Raj, Niti and all the group who organized all this. Without my knowledge in the background, all these many pictures which are being presented today, I would like to keep a copy because I don't keep pictures, I don't have much stock except a few which I would like to share with you. Nasreen, thanks for coming. She was with us in ICGEB. We have jointly written some paper also together. So there are many of them who have been co-authors with us at ICGEB as well at JNU. Thank you everybody for whatever you said. This is briefly a glimpse of academic journey, but I'm not going to talk science except taking you through to the people whom I wish to have gratitude to all my mentors, colleagues and students in the last 50 years. It is not easy to compile everything, but I'm rushing through this one. So I started, as was mentioned, my master's degree in Jammu Kashmir University, moved to Delhi University to do my PhD. From there on, I was at JNU as assistant professor. I was taken there and then went to, I didn't want you to go, Meena. No, it's not that. Huh? अच्छा वो फ्रंट में तो और मुश्किल हो गया. So from JNU, I spent almost 20 years in JNU, but in between I moved to different places, basically to enrich my knowledge, to enrich my skills. That was something in the background that I must go along with the new technologies which are coming up. So we went to Max Planck Institute in Cologne. From there also I went to UT Austin. Stan Rue, you just heard him, spent more than 1.5 years with him isolating phytochromes in the dark rooms. That's what we learned from Stan, that you have to come 5:30 in the morning. If you have to start, those days there was no genes for phytochromes. So you could not purify proteins, you have to purify yourself. And the whole purification had to be done in darkness and in cold room. And that was the tough thing which we had to do. And it had to be finished in one day. Because you can't keep phytochrome, it is labile. And so we had to start 5 in the morning with 4 kgs of Avena coleoptiles, grinding them and going through the whole process. I don't want to get into details, but that was the kind of training which we were getting. I went to USDA, Avtar Matu just talked about it. Marvin Edelman, who was in Weizmann Institute, used to come there at that place. So they are the kind of mentors which we had. I spent some time in University of Munich. Ralph, who just gave his talk here, is sitting in the back. A lot of work together. And also Professor Herman. Herman was also a tough taskmaster and wanted things to be done on time. So these were kind of destinations which I had while I was in JNU, and finally moved to ICGEB and back to JNU and back to ICGEB. The arrow is showing that I'm traveling around JNU and ICGEB. And all these people really were very influential in my career, and especially my PhD boss, Professor Satish Maheshwari, who made a big change in my career. when I came to JNU, I got a lot of support from School of Life Science colleagues who were already there or who joined there. And many of the people you must recognize in the first row. This was the MPhil PhD batch. Dr. Rameshwar Sharma is in the back. Then Neera is around there. And other people around. Neera, you can recognize all those people around there. And these are the professors sitting in the front. Professor Mohanty, Ashish Datta, and Gauri Singhal. And I don't have, this is the marker? I don't. This is Santosh Mukherjee about Ete was talking about, and this was our first Vice Chancellor, Professor Partha Sarthi, and Shipra Mukherjee, Keshwan, and all these people, colleagues, we have been there. When I first joined, I was interviewed by Partha Sarthi, and he was selected me. And after selection, you go there. He wanted to meet me, and he said, "We have selected you not that you have done anything great science, but I selected you because I see a potential in you." I was surprised, he said, "I can see through that you will do something good in future years at times." Thank you, Partha Sarthi, for selecting me. So all these students were the first best students, and the teachers there, a great help to me. The backbone of my research has been large number of students, postdocs. Whatever names came to my mind. Hi, Jagdish, please come here. And Neera's granddaughters, they are as tall as Jagdish, With Neera and Jagdish, it has been a family kind of ties which we had. So these are all the students, postdocs, which have helped me all in my career. And I used to share our lab with Shipra Mukherjee and many of our students. It was like one lab, all the students, whether registered with Shipra or with us, we were doing all the work together. And Rajshekhar, I thought I would pick up this. This was his PhD thesis. I have kept theses of almost all the students in home now. One Meena is upset that you're occupying so much of space with this. But I picked up Raj thesis because nowadays in many journals you see that they give you the abstract, which is a graphic abstract in the abstract. Now this is a recent trend, last few years it is coming up. The Raj actually developed this long back. The graphic abstract of PhD thesis was given on the title. So you're the originator of this concept, which has been taken over by different journals now. And we had a great PR lab, plant research lab in JNU. A lot of people, Raj is here, standing here, Neera, and all these people are around here. And we used to have a lot of meetings, organizations. I may not be having pictures for many people. These are also, I was trying to look into my drawers if I can find something. I don't very good in keeping these pictures, but it was a very vibrant lab that we had. One person I would like to thank at this occasion is Professor P.N. Srivastava, who was our Dean and also the Vice Chancellor. He helped me in many, many ways in my career. I cannot go through all that, but he was a wonderful Vice Chancellor who always said that I'm not subservient to the rules of the government. I go by what the situation at the spot is. And he sanctioned me 40,000 rupees out of my Provident Fund account, which was not allowed by the government, but to buy a car. I said, I don't have money to buy a car. I had booked for Maruti car. He said, कि मैं इसमें से दे देता हूँ. The finance officer said that we cannot allow it. He said he will return it. It's on my guarantee, and he sanctioned. So he was that kind of person who would try to help. So these are some broad areas people have already mentioned. My early days on plant tissue culture at JNU, we worked on light regulation of enzymes. Many people mentioned about it. When I moved to ICGEB, we went to abiotic stresses, gene function relations, and mainly glycolysis pathway, about which Arun talked about. And post-retirement, we're talking about light and stress signaling, stress priming mediated memory mechanisms. And recently got interested into these prime-like proteins. It's a very few papers on this. Only 2 or 3 papers are now in plant sciences on prime, and one of that is our paper. And more work is being done by some of the people in the lab now. So this was the earlier concept. We also had started, Neera, remember, started with Arabidopsis research. She used to grow those Arabidopsis, trying to find out certain mutants crossing those protoplast culture and so on. And it was interesting that in one of his review article, Elliot Merowitz, when he talks about the prehistory and history of Arabidopsis research, this was the conference I attended in Frankfurt. He gave that picture, not that I've done much contribution except to be standing there, but I made a presentation on Arabidopsis. So in Arabidopsis history, this record has been there. And one of the students who was with Shipra Monjan, we did publish a 5-volume series on haploid culture, which was the research done by Satish Maheshwari in Delhi University, one of the first. And these books were written in honor of Professor Satish Maheshwari. This 5-volume series was brought out. So basically at JNU we are working on light and nutrient at one hand, light on other, how these interact in order to regulate these developmental processes, the signals, kinases and all that. And Raj worked in chlorophyll, carotenoid development pathways and so on and so forth. These were the kind of research areas which we were doing. Then I moved to ICGEB. I was due to Dr. Tiwari, the person who first isolated this chloroplast DNA and all that replication mechanisms. He brought me here. Hiraj has worked with him as postdoc and many others here. And this Professor Maheshwari. And I received a lot of support from the Director General, directors of ICGEB at one time or the other. Barale here was very, very helpful. This is Dr. Chauhan, who has been director here for many years. We went with him together to Chile, where there was some meeting. And Dinkar and Mauro Jaka, I think they have been the Director General, very, very helpful. And Louis Banks now. They're all been very helpful for my stay in ICGEB, and I'm sure Dr. Sonti will help me too. Thank you so much. It's very nice that plant scientist has come back to this institute to head this place. This PMB group, many people showed this picture, was a very, very nice one at second floor. We used to call it second floor ICGEB. Many parties together, whenever there was a new paper published, we'll celebrate that one. This is not happening anymore because they have too many papers now. So how many times to celebrate? Itna samosa khana padega ki badi mushkil ho jayegi. So I think those days when our first plant journal paper came, that was the first from the country. It was a great celebration when Veena was around. And, so these are the kind of research work which we were doing. Not anything on perception, but basically we are working on signaling what kind of omics changes takes place and how these help the plants to adapt to different environmental conditions. We had isolated many genes, working in collaboration with my colleagues, very nice persons. In our PMB, we had a physicist, we had a biochemist, we had a geneticist, M.K. Reddy is here, he was here. And all these people brought different kinds of expertise within the group. And this was the pathway on which we continued further for a long time. Arun has already talked about it, but I want to acknowledge some of the students here. When we started this work, actually it was started in JNU. There was only one publication in Glycolysis research in plant science. And then Ramaswamy took it over, and many other students followed on this. We were loners for many times. Suman Bagga, Tarak Chakravarti, who is no more with us now. Paulus came from Germany to spend some time in our laboratory to characterize this one. And many of these people who spend a lot of time, Renu talked about, Renu worked on this purification of this glycolysis. And this continued for many years, and people thought initially my projects were also rejected because they said this pathway has no role at all in plant systems. But now I was so happy this year that almost like 35 papers have already appeared on glycolysis research in plant sciences, including Arun Sharma's now recent paper in Plant Cell Environment he has published. Post-retirement, I got SERB Distinguished Fellowship, and thanks to ICGEB for senior emeritus scientist positions. Some funding came from there. And these are some young groups whom you saw that they were talking in the laboratory, something about me. I couldn't hear many of those things, a few of those together. And these are the young group which is around there. Some of them are around, some have left. Very, very hard-working people. But I have collaborations. Thanks, Sneh Neeti, some collaborations going on with her. A small collaborative project was done with Shudeep at NIT Durgapur. A few pictures I have. Raj, you remember, I visited you a long back. Your daughters are grown up. This is a very, very old picture. Stan visited us for the international meeting, his wife and Professor Herman around here. This is Jakobsen from Germany. He was with me in Germany, but later on in Netherlands, who worked in potato. So he visited us. Reena Das visited him, and I don't have too many pictures, but many pictures which you have, I would like to have them with me. And then back to JNU, as they said, Kaveri Hostel. This was the news. Kaveri Hostel warden Sopori returns to take charge of JNU. And I was so scared, when I joined JNU. I said, "Ab dharna varna to bahut hoga yahan pe. They will have demonstration." But anyway, I really express my gratitude to all the JNU faculty, to staff, the students, and the cooperation and support we got. I was really inspired by the students, this especially abled students. They were a great source of inspiration for this. I'm so sure Akansh, he came here, many of them. I had a special time for them and tried to do whatever best I could do in terms of developing the campus, providing facilities and so on and so forth. Many of them are still in contact with me, and that is the good thing which I feel happy about. And next, चलने का क्या हो गया? नीति, इसको देखो क्या हो गया? It doesn't want me to come back. And this was a kind of experiences which I had. One of the experience was to meet different kind of people who also give me a sense of confidence. With Pranab Mukherjee, I could tell stories about Pranab Mukherjee because we started having a lot of interactions together. He took me along with him for his foreign trips 2-3 times, that he should come along with me in signing certain documents and all that with the universities. And a great personality. Even after having spent his time with the ministers and the government officials, when he would return to the hotel, evening he will call you, "Please come to my room, tell me what have you done today." So he was like a hard taskmaster to see what we're going to the universities, what we learned there, and so on and so forth. And a few hours that I spent with the German Chancellor, this President of Germany, very, very interesting person, and the kind of way he talked about it. So what I learned, life lessons, quickly go through these. This is what Professor Maheshwari used to tell me, that you must read about the history of science all the time. When you read a paper, you must learn about the author. So he said, you always talk about the science, you never talk about the person who did the science. And therefore, reading through all these historical persons, these are the kind of, he said the qualities which one should have to be a good scientist. I don't want to read 3, all of you have these qualities. Good? Really? Yeah. The other interesting quality you should have is retain the childlike behavior. This is what I like about what she said, Barbara McClintock. She never published papers in high-impact journals, but she got the Nobel Prize. And she was saying that I was just so interested in what I was doing, I could hardly wait to get up in the morning and get at it. One of my friends, a geneticist, said I was a child because only children can't wait to get up in the morning to get at what you want to do. So that's the kind of things. You have to rush to the lab. I have set up an experiment in the night, what happened in the morning. So that curiosity of going back to the lab and looking at things. Some other useful advice I received from many mentors. One is think independently and frequently. Your currency is good scientific papers. As Benjamin Franklin writes, either write something worth reading or do something worth writing. And learn from failures and overcome fears. And that's our problem is sometimes if our experiments don't succeed, we get frustrated. And my Professor Herman once said that if your experiment doesn't fail, you are not a good scientist, you will never learn anything about it. Because next time your student's experiment fails, you will not be able to explain why it failed. Other thing Professor Maheshwari taught us, read widely, stay ahead. And those days there was no online, we had to go to the library and read, and he had given me 30 topics on which I must be updated. Besides my applied research and tissue culture research, and there were seminars on that time. When we started our career, there was only one nature, and today this is the situation. And in science there are 11,000 journals. How do you keep track of it? You don't have to keep track of everything, but you must read beyond your own research area. That becomes very, very important. Again, stuck up. I am touching something wrong. Niti, few minutes more. The other thing is that you must keep updating one's skills, because technology is revolutionary. There's many things which are happening. We started with gene cloning long back in 90s. In fact, the first gene in this country was cloned in 1984 by Padyati in Indian Institute of Science, Bangalore. So that late. And only when CPMB came in GNU and Delhi University, some work on molecular biology started. But now things have moved very, very fast. Renu talked about proteomics and sequencing and so on and so forth. So you have to follow developments in technology. This advice I received and I'm passing on further. Things are moving very, very fast in different directions, especially microscopic technologies. My first paper was on chromosomes from Kashmir University, cytomorphological investigations in Chlorophytum capiens. That was my MSc project on which we got a paper in Indian Journal of Horticulture. So moving from Indian Journal of Horticulture to PNS and others, it's been a long journey for me. And we used to do this chromosome count. And now this chromosome biology, the way it has moved due to these technologies, and the recent paper which came, surprising arrangement of DNA in the cell's nucleus by imaging technology. The DNA is not floating around, it's on the sides here somewhere. So when you talk about gene expression and other things, you have to look at what the physical location of these structures are there. This is something which we have to keep ourselves in mind. The other advice, follow research project that has originated from you, and the project can be basic or applied, and this is the evolution of an intellectual freedom. When you are, before joining PhD, you say, I will like to do research, whatever I want to do. When you become graduate student, you say, I'm going to do research, whatever my professor tells me to do. Move forward when you are assistant professor and professor, that you will do research only from where you get the grant. So whosoever granting agency gives you, then you get into this. And finally you become also grant committee members and all that. You don't know what to do with that. And he says finally, research in peace. So you are celebrating my 75, I'm somewhere here. So I have to do research in peace. Other thing is foster collaborations. This is very, very important. Nowadays, as I was telling, technology and literature is moving so fast. You must collaborate. We had many Indo projects, Indo-Belarus project, Indo-Israel project, and with Jaco we had some projects, and many other collaborators around here. The parties around with Kailash, we had done Assis, and many other people. This is Alok, when he visited us in Germany, and this is Nupur. No, she's sitting here. He's around here, and Meena. And this is mother of the Nobel laureate Venki, who came to Hamburg to attend the meeting. And with this collaboration, some write-ups also came up. Meenakshi helped me into doing this. Meenakshi is here. Yeah, so we have to revise this. So you're saying you don't have any work, so we have something needs to be done further. Okay, the other thing is, which is missing nowadays, many people, is to attend seminars. Professor Maheshwari and many others in Germany, what I learned is must attend seminars. You imbibe many, even if it is not in your area, try as much as you can attend those seminars. So my take finally is 3 T's which I want people, time. You must respect and manage time very well. Talent within yourself and also within your students, you must nurture, build their skills. Teacher has to be a good listener, compassionate yet result oriented. You can't be only compassionate, You must see that it works out. And the 3 Ds, which are discipline, becomes absolutely important. Discipline, mind, and your action on your experiments. Duty, perform to the best of your abilities you can do. And of course, dream. Your project should be dream big. Not only dream big, make it happen. And above all, think beyond self, value human relations. This is what Swami Muktanandji, he wrote to me once. Relationships are like bank account. You can withdraw to the extent you have deposited. And after retirement, moving into the domain of awareness and consciousness, this was induced in me by Dalai Lama when I met him, and he asked me to conduct a seminar on this, on quantum physics and consciousness. And I've been reading about it from childhood. I had some of those exposures. But I thought I should also give a talk once to Tibet House, do plants have consciousness? And reading about this, although we have been talking about perception and awareness, I finally got this book compiled, which talks about awareness in the plants, perception of the environment, molecular computing which happens within the plant systems, how plants communicate, concepts of cognition, experience and memory mechanisms in plant, and finally addressing the phenomenon of death in plants. The huge trees, sometimes I feel the death doesn't occur there. You know, the 3,000 years, 2,000 years, how do these suns survive or these trees survive for such long? These are the kind of imaginations which people have. Most of the people who wrote in this book are my former students, mentors, and Ramesh Sonti also contributed an article on this book and others. So this was my acknowledgement to the students who contributed in my academic journey as the form of a book. Another one I have to do because there are many more who needs to be taken into that. So this last is moving into RIP. Somebody sent me recently because my aunt was a Shivaite saint. She passed away. On passing of, this was written by Bettina Baumer, who was her disciple from Germany, from Austria. She wrote, "The body is an offering. The human is an offering into the divine. The body is an offering into the fire of I consciousness. Gravity is an offering into the lightness of grace. Time is an offering into the timelessness. Sounding is an offering into the silence. Movement is an offering into the stillness." And I feel the last two are plant. They work in silence and they are still, yet they perform their functions. Lastly, thank you family for being with me all these years. Support Meena. Never complained, as I've been always mentioning. My being away many times. So thank you. And thank you everyone for being a part of my teaching and learning experience all these 50 years. Thank you everybody to be here.
So we are joined by a few other people from JNU. They're finding it difficult to climb down the stairs. So some of them want to say a word. Somebody wants to say something? Oh, she wants to come and say.
Okay, thank you. So, very good evening to all of you. Okay.
Dear Sopori sir, all dignitaries, our colleagues, peers, ladies and gentlemen, such an overwhelming moment. I fall short of words. Sir has been a great inspiration for all of us, and so has he been in my life. I've already expressed this in the video, and I ensure that I wear the same jacket so sir identifies me. So, yeah. I'm Gunjan. I have been a JNU student. I'm from Center for English Studies, and I graduated recently. I completed my PhD, thanks to sir. I remember so many things. I have to say so many things. You guys tell me how much time do I have? I mean, 2 minutes, 3 minutes? Okay, I'll wind up immediately. I just wanted to say thanks to sir for so many things. You know, how people see disability, for instance, whenever the idea of disability comes, there's a hushed silence, there's a silence, pin drop silence, as if something disastrous has happened. Right, when I was walking upstairs, like everybody was quiet, what is going to happen? Is she going to fall down? So this was never the case with sir. He was like, let's talk face to face, dharna karo, I don't mind. And I also remember, and as sir was saying that, you know, only compassion doesn't work. You have to also perform. And this I have learned the hard way. It was a great time for me. Whenever we would go with any project, he would appreciate us. That was the time when it was thanks to sir that all of us got laptop for the first time. All visually impaired people got laptop for the first time, and sir was not at all ignorant about things like screen reader. It's like, is JAWS installed? I was really taken aback. He said, is JAWS installed in your laptop? I don't know how many of you knows the meaning of JAWS. That is Job Access with Speech. That is for Windows. And obviously we have voiceover. One incident that I would like to quote about sir is that once we organized a function, a program, and I was comparing. So in a heat of moment, suddenly I said, now may I allow the Vice Chancellor to come on stage and share. And then I immediately corrected myself, and then obviously I was scared after that, and then he came, yes. So since I've been allowed, I'd like to, you know, begin my speech. So was his humor, and so was his forgiving nature, and after that he patted me, saying that good job. I mean, sir has always been like that. I'm sure sir has so many things, and he might not remember me, but I have fond memories. And I have learned a lot, we have gained a lot, we have protested, we have won so many battles, and with sir's blessing, I think I will continue to win many battles and finally the war. Thank you, sir. How do I get this? Thank you so much, sir. I hope you remember.
I remember all of you.
Yes, sir.
Your pictures are with me in my home.
Thank you so much, sir. Thank you so much.
So now that it has triggered the felicitation process, may we request Raj and the other endowment committee members to also do the job. Yeah. Is there anybody else who wants to speak? Any of you? Sir. So after this we'll continue the celebrations in the guest house. So there would be a trail for the cake and request you all to follow it. Please. Yeah, and another message that there are still lot more videos which could play throughout the night. So we'll put them online and we'll share the link, and then you can keep watching them, because it's really going to take a lot of time. So many people have sent in their wishes. So can we move towards the guest house for the cake cutting and celebrations, because auditorium it will be difficult for us. Thank you. Can we all please move towards the guest house for the cake cutting?
This one, crispy item.
ये मैम हाँ नहीं मैम 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 मैं यहाँ इम्पोर्टेंट दोनों है आ जाइए कैंडल में जला दो रेड कैट्स वेटिंग फॉर मैच
मेरा ये नहीं खिलाया आपको लाइट but I guess different ways of looking. Sorry.
वो से नहीं होता हाथों से खिलाना होगा
फिनिश हो गया, लॉक कर दूं? लाइट बंद कर दूं? हो गया ठीक है।
बस वही
बनने क्या भी करूं?
वो नहीं है, फोन ही नहीं उठाते, पूछ लो ना ये। हेलो सर, अब बंद कर देना। हाँ एक मिनट बोल दिया बंद।
कोई बात कुछ
ठीक है बंद। स्टॉप।
बंद कर दूं लाइट? तो ये फोटो भाई यूट्यूब लाइव चल रहा है।